convergency, when we talk about reliance. And one should always keep in mind that convergency is a harmonization, it's an alignment process, and a process in which the health authorities use common analysis criteria and common technical specifications that are required for the approval and monitoring of products. But that does not mean an automatic recognition or the acceptance of criteria that are imposed by other authorities. So this is an important starting point when we talk about convergency. Besides that, when we think about today's scenario, which is an scenario that Anvisa and many others authorities face today, which is the issue related to human resources. So how can we balance the limited amount of human resources that we have with this greater universe of processes that we need to regulate? And when I'm talking about processes, I'm not just talking about this analysis queue, because this is what we need to do. We need to deliver products that are safe and with efficacy to the population. But that also requires regulatory processes that are more complex, that follow good regulatory practices, but it also require more time and work from us. So convergency brings one possibility to optimize our analysis and reduce the time they take. And with that, we can bring access to products that will have the same level of quality, but at the same time will reach the market faster. The whole world has been working in different convergency strategies, and I think these events and discussion forums where Anvisa goes to, which are global forums where Anvisa can discuss with European and American, Australian, Canadian authorities on equal basis. And we have a voice in these discussion arenas, and that allows us to bring similar criteria and lead our analysis in a similar way to these other authorities. It is important for us to know what we have in-house, so then we can think about different strategies to tackle this. And in the end of my presentation, I have a provocative question related to innovation and how we can balance these issues of human resources and the processes that we need to analyze, innovation and access. So it is important to know our own scenario so we can adopt strategies that are effective for Brazil. Because we have a unique reality that requires its own specific approaches, so we have effective strategies in dealing with our demands. So when we look at the regulated medical devices in the country, we have almost 70% of imported products. And that makes us think, because I need to think about strategies for dealing with these imported products, which are part of the majority of the products that we look at. But we also have 30%, and again, we have more than 80,000 medical devices analyzed in Brazil, and 30% of them are national. And this number of products of manufacturing in Brazil is only going to grow. So how can I adopt regulatory strategies to foster this market so I can have not only strategies to bring imported products to Brazil faster, but at the same time strategies that will foster national production and bring innovation and product manufacturing to Brazil that will be regulated by Anvisa and then will be exported to other markets as well. So this is just some food for thought, meaning that we need strategies not only for convergency and reliance, but I also need strategies to deal with the products that are manufactured here. This has to do with class of risk or risk class. Almost 80% of products are classes 1 and 2. And when we talk about reliance strategies, we have been discussing them to a great extent. There will be a specific standard to bring this criteria for medical devices. And again, we are talking about classes 3 and 4. These are the products with the greatest level of risk. So by using this strategy, I will cover part of this that generates a great amount of demand, but at the same time, it only represents 20% of what we have to be analyzed. These are the deadlines that we have had for the past few years for regulating these products depending on their class. And it's important to notice that many times the regulatory interventions that we make have an impact on these deadlines. And this is a moment where the waiting lists, they grow not only because of the regulatory work, but also because of our lack of human resources. The directors of our agency have been raising this issue quite often, the shortage of labor that we have, and how we can continue to work on our demands considering this lack of people. So we start to see this increase in our deadlines that will require us to adopt a strategy. And one of them is the reliance strategy that will be discussed soon, one that we hope will generate an impact on these terms that are part of the scenario we have today.
So as I have mentioned, we are dealing with classes 3 and 4. So we're dealing with a smaller part of our demands, but at the same time, it's an expensive one. So this is a strategy that we're dealing with, considering the gains in other areas, because we will have specific staff to deal with products with class 1 and 2 and other regulatory demands. We will have more people to deal with other standards and other themes that the agency needs to work with. We have an important experience considering medications, and I would like to mention specifically biologicals, which was an area at Anvisa that started adopting reliance strategies back in 2018. And the result that they have today is a reduction in by 80% of time of analysis of the dossiers. So the result that we get both from the industry and also employees, that is a win-win relationship, because there's a gain of 70% in time to analyze strategies. And so the industry has access to that, not only the industry that submitted the processes in that format, but also people who are in the lines that will not use reliance strategies, but are able to have faster analysis because there is dedicated personnel to that. This is the efforts trying to take care of everything and trying to, conciliate that with new submissions. The number is increasing exponentially, and the decrease of personnel. So we're thinking of that scenario and trying to come up with strategies to optimize our work, even facing the scenario that we have. We participate at different fora regarding medical devices. So the IMDRF, the MDSAP, that already promoted good gains regarding the certification of good manufacturing practices. And so that shows the increase of the number of plants participating, and this is the number of certificates issued. So here you see that there's an increase per year, and we did not observe any decrease in quality or in surveillance, post-marketing surveillance of these devices using this convergence and reliance strategies adopted by the agency. So that leads us to believe that it is possible to maintain the quality of our analysis using the regulatory trust. So we're able to trust the analysis conducted by other agencies that use similar criteria as Brazil. So we're able to trust their analysis in order to improve and optimize our own processes. It is always important to strengthen the surveillance. That's what we always say. Every time the agency needs to flexibilize criteria, We also need to strengthen the post record so that we can surveillance, monitor the product and the market in order to actually assess the quality and the maintenance of the quality of the product. So this is the definition of reliance. And just like I mentioned before, reliance is not something that removes the autonomy of the agency. So Anvisa continues to have autonomy to make their decisions considering data submitted by the company and also considering the Brazilian reality. Many times we see medical devices submitted in Brazil and they might not be approved in the same conditions in another country because we have our own reality. We have the national health system, the SUS, and we see that some products are incorporated in clinical protocols that are not available in other countries. So we will always need to assess that considering the reality in Brazil. So it's no use approving a cancer product for second line here if we do not have the first line approved here in Brazil. So that is why we need to maintain our autonomy and consider the specificity of our country in our assessments. At Anvisa, we already have the general regulation, which is the umbrella standard, which is our RDC 741 from 2022. And that standard lays out the general criteria. Based on that standard, we establish specific criteria according to the category of the product. So I have criteria for medications, for medical devices, for cosmetics, and for agricultural products. We have been working on these specifications, on these criteria that will be defined for medical devices. What are the conditions that are applied when we deal with reliance? First of all, we have to have an agreement for information exchange with the other regulatory authority. So if I have an agreement with FDA, I can exchange information to help with the analysis and help assess the risk and the benefits of that product. So it's important to have those agreements with other regulatory authorities. This proposal also involves medical devices of classes 3 and 4, which are the ones of higher risk, and the product must be essentially the same. So regarding all standards, we cannot submit a product in Brazil that it's different from the product approved by another agency. So the product submitted to our analysis has to have the same indications of use, the same places of manufacturing, and the same regulatory version. That product must be then submitted with considering that same regulatory status in the reference country. The Brazilian requirements regarding labeling must be met. So this is another important aspect. When we talk about that reliance, we're not letting go any regulatory requirement. The dossier is the same. We will only optimize the analysis and we will not analyze completely some of the documents because we trust the analysis that was previously conducted by another agency that has similar criteria as ours. So the documents are the same that we already require. Other than that, we can always do a complete analysis, either because of internal strategies, because of audits that randomly require some documents to be analyzed completely, or when a technician identifies that there's a need for a detailed analysis of that process. The agency is always, able to question documents that were submitted as well. The authorities that are laid out in the proposal are the Australian, Canada, Japan, and US agencies. So whenever we start those reliance strategies, we start in a conservative way using regulatory authorities that are actually a reference, considering the analysis of that category of products. So regarding medications, we started with the European and American authorities, so EMA and FDA. So we start with few authorities, and once the standard becomes effective, then we evaluate the surveillance so that we are able to broaden the universe of authorities involved. And this is a food for thought regarding innovation. Just like I mentioned before, we talk about reliance and regulatory convergence mechanisms, but at the same time that we're dealing with these imported products to optimize our analysis, we have to have strategies to welcome innovation. So that cannot be restricted to optimization. We have to be able to welcome innovation. We have to catch up with innovation because the innovation in the industry, specifically when we're talking about medical devices, we have to think about coming up with a standard that is flexible to welcome that innovation, but at the same time guarantees the safety, security, and efficacy of that medical device. So whenever we're talking about Brazil, we see that the country needs to advance regarding innovation, and that is due to several factors. We need to advance in clinical studies, for example, and then we have the standard for clinical studies that it's being elaborated by the agency, which will come up soon. And this is a strategy to simplify and debureaucratize the process. This is a country that is able to conduct clinical studies and will advance for sure in innovation. The interaction between industry and academia is also something that needs to grow. We see there's a lot of, when we look at the number of researchers per inhabitant, or in the pharmaceutical industry, or in the medical devices industry, it's a limited number of researchers, so that contributes in that universe, to that universe of innovation. So why is it important to be able to deal with innovation and to have standards that allow the entrance of innovative products in Brazil? Because innovation will represent the increase of quality of life. So these are products that offer significant results and outcomes for patients in terms of treatment and medications. It increases the competitiveness in the market, improves the internal capacities, the quality, and patient satisfaction. But the challenge is how to regulate innovation. Our Department of Medical Devices has been working with a request for proposal at Anvisa so that we are able to monitor, just like it's done in the US, FDA. The FDA has a group that monitors and survey the development of medical devices since the development till entrance in the market. So we have a request for proposal at Anvisa as well, so that we are able to welcome that type of innovation. So they will have a special place in that request for proposal, and that has to deal with imported products as well, because whenever we are able to regulate that product, our standard might not meet the criteria of that product. And since it's a product that it's monitored by the agency, since it's design, then we will be able to create a specific criteria so that that product can enter the market. So our role is to create a safe environment for technological development, to update the regulations considering the new technological realities, evaluate the safety and efficacy of products to become a national reference, and participate in a proactive way in the evolution process in the industry, and promote cooperation and the exchange of information among other agencies. So we have been trying to work on that path so that we are updated considering the incorporation of innovation in our country. Here are some innovative technologies and some recent challenges that we have faced, both in the areas of medications and medical devices. These are innovative products that we had to update ourselves considering the technical and scientific criteria to analyze these disruptive technologies so that they could reach the general population. So implantable devices, intraocular devices, synthetic and biological drugs, cardiac valves, gene therapy, and mobile apps for monitoring patient levels. So these are challenges that always make us question our monitoring capabilities, and that device has to reach the market at the same time that the technology is being used in other countries. So considering all these challenges, how do we have to work? That is through intelligent regulation with regulatory convergence, balancing regulatory convergence, imported products with incentives, strategies to the national industry so that we can welcome Brazilian products so that they are regulated in other markets as well. A regulatory sandbox, which is a specific strategy considering innovation. It allows us to create a controlled environment where we will monitor that standard and its outcomes in specific situations. Promotion of access to innovation, tools of AI and information technology. This will increasingly help us with the analysis and also contribute to this scenario of reducing the number of people so that we can have faster analysis through IT tools. And last but not least, the reliance practices without losing sight of national products that also need specificities in our regulation. That would be all for today. Thank you so much for having me, and I'm available if you have any questions. We would like to thank Daniela Mareco Cerqueira for her presentation. She's the secretary of the Chamber of Medications at Anvisa. I would like to acknowledge Patricia Briley for her presence here. Now we are going to talk about the OMS vision for a harmonized environment and improvement of the regulation of medical technology. So now I would like to invite Augusto Geyer, the General Manager of Technology at Anvisa. To discuss the theme, we have an online panelist. We have Susie Braniff, Technical Officer from WHO. Susie, can you hear us?
Yes, I can hear you.
We also have Daniel Yoon, Regulatory Advisor for Health Canada. We also welcome you on board. Daniel Yoon, do you hear us?
Yes, I do.
Can
Thank
we
you.
hear him? So Susie Breniff, Technical Officer at WHO, do you hear us? Ms. Breniff?
Yes, I do. Yes, I do. Thank you very
Thank
much
you.
for letting me participate today.
We would also like to invite Sima Vias, Director of Regulatory Issues at Medtronic Affairs. Sima Vias, do you hear us?
Yes, I do. Good morning, everyone. It's a pleasure to be here.
Augusto, the floor is yours. Good morning, Susie. Good morning, Sima and Daniel. Good morning, everyone here in the room and also our audience online. So the idea of this panel is for us to have a general discussion about the harmonization and regulatory convergence issues. So how does WHO deal with that? Specifically talking about its authorization processes and qualification of products. Daniel Yoon will also comment about a tool that has been adopted both by Canada and US. And that tool is called eStar. It's a tool that allows companies to present their submissions in one single document structure, so to speak. And so this pilot project has been having a fair amount of success in those countries. And so Daniel Yoon will talk a little bit about how this has been adopted. And also we have Sima Vias to talk a little bit about the use of that eStar tool by the industry that submits these types of documents to these two countries that have been conducting this pilot.
So once again, good morning, Susie, Daniel, and Sima. Welcome again to our International Seminar on Medical Devices. And right now, I will hand the floor to Susie Breniff so she can conduct her presentation about the WHO's view on a harmonized regulatory environment and how to improve access to medical technology. Susie, the floor is yours.
Susie, just a second.
Susie, just a second. We are having a technical problem. We cannot hear you. Just a minute, please.
Okay, I think I've unmuted myself now.
Ok. O som
Okay, so,
agora para ela?
Susie, I think we're ready again.
okay, thank you. Sorry, the problem was on my side, I think. I wanted to say greetings from Geneva and thank you for allowing me to join remotely today. I want to talk a little bit about the pre-qualification process for IVDs and the ways we have worked to introduce reliance mechanisms into this process so that we have a more efficient system. So today I will just begin with an overview of the way that we assess IVDs for pre-qualification, but then also look into the two areas where we are using reliance strategies to assist with this work. The first being the use of an abridged assessment, and the second being the collaborative registration procedure that we conduct with other NRAs who are member states of WHO. So to begin, the pre-qualification process for IVDs has officially been in place since 2010, but WHO has a long history of evaluating diagnostic tests, beginning largely with the HIV pandemic back in the 80s. And over time, we have expanded the scope of the assessments we conduct. So in the blue box I have up on the screen at the moment, you can see the different disease areas where there is a pre-qualification assessment available for different products. In the lighter blue text at the bottom with the stars, these are some more areas that are coming into scope during the course of this year, and we continue to expand the scope of pre-qualification based on the requests from our member states and stakeholders. And the goal of our program is to promote the access to safe, appropriate and affordable IVDs of good quality. At the moment, we have 110 different products that have been pre-qualified, and we maintain the list of products on our website, and whenever a new product is added, we will update the list accordingly. The assessments that we conduct under pre-qualification, we have worked really hard to make sure that they are in line with international best practice. So when we develop our technical specifications, we take into consideration ISO standards and also guidance from IMDRF and its predecessor, the Global Harmonization Task Force. These go into the development of our technical specification series documents that we make available to manufacturers so that they can prepare their product dossier for us. And we also have a set of technical guidance series that cover some general areas that we would like manufacturers to consider when they are planning their validation studies to be able to demonstrate the performance of their IVDs. The pre-qualification assessment consists of 3 different components. There is the review of a product dossier, there is a laboratory performance evaluation, and also a manufacturing site inspection that covers all sites that are related to that product. To the side, I've also mentioned a labelling review. So this is conducted at the end of those 3 different assessments to make sure that the information that's going to the end user is clear and also accurately represents the performance of the product. I'll quickly go over those 3 different elements of the assessment. So the review of the product dossier is looking at the manufacturer's data that they submit to us. And we have a subject matter expert go through a product dossier. And what they are looking for is they will conduct an evaluation of the relevance of the data that has been provided. They're looking for credible data that supports the manufacturer's claims of quality, safety, and performance. And they will also consider whether the studies that have been performed are appropriate and well designed. For the product dossier, they're taking into consideration if there's evidence of a complete body of evidence for that product. They're also looking at how accurate that data is and has it been consistently collected. So they need to consider, is there information that they can see from the initial product design through the way that product has been validated and is now being manufactured, the quality control and release onto market, that all of these things support the quality of the product. The 2 main questions that they will provide input on are, is there evidence the technical specifications that WHO has prepared for that type of product, have they been met? And we have different TSS documents that are tailored to the specific analyte and assay type. These documents, as I mentioned, are aligned with international requirements, but are also made to meet the WHO requirements for a product in the different member states. We are also very interested to see if the manufacturer has considered the use of its product in resource-limited settings. For the performance evaluation, we are conducting an independent verification of the performance of the IVD that has been submitted. And the product will be challenged with a focus on its use in resource-limited settings and also in the context of the relevant WHO guidelines for that type of product. In each case, a standard pre-qualification protocol will be followed for the evaluation, and this data set is intended to complement the manufacturer's verification and validation data. These laboratory evaluations take place in either a WHO collaborating centre or a designated performance evaluation laboratory. And we have 14 different PELs that we work with that are located in different areas around the world. As part of the evaluation, the specimen panels that will be used have been designed to represent a global population. And finally, I'll quickly mention the manufacturing site inspection. This is done by a dedicated team within WHO called the inspections team. And they are going on site to look for evidence of a fully implemented quality management system that meets international standards. Part of the considerations that they will have is whether the product itself is going to be robust enough to be used across the many different member states. And they need to see that product in manufacture during the site inspection. The other key piece of information they are gathering is whether the manufacturer has sufficient capacity to ensure reliable delivery, as if that product is pre-qualified, they may receive an increased number of orders for the product, and we need to be assured that the quality will be maintained if this occurs. So the pre-qualification decision depends on a successful outcome in all three areas of the assessment. A product will be added to the PQ list if it meets all of the requirements in these three areas. We do make some distinction between critical versus non-critical issues, and in some cases, corrective actions or commitments to pre-qualification will be added with timelines to give the manufacturer time to demonstrate that they are fully compliant. The final outcome will depend on, for dossier assessment, the acceptance of an action plan for any outstanding issues, the meeting of acceptance criteria during the laboratory evaluation, and the results of the inspection and the acceptance of an action plan by the inspections team if necessary. At this point in time, the final labelling review is conducted, and if the manufacturer agrees with WHO's labelling suggestions, then the public report is prepared and the product is added to the PQ list and put up on the web. At this point, it becomes eligible for WHO and UN procurement. So that's the overview of the way we assess the products. Now I want to switch to how we use reliance within that process. And I'm sure everyone here is aware what the definition of reliance is, where an authority in one jurisdiction gives significant weight to the assessments conducted by another regulatory authority or trusted institution to help it reach its own decision. So part of the components of a good reliance system is that it provides a platform for international cooperation to ensure the safety and quality and performance of the products when they're used locally. Another benefit of using a reliance strategy is the efficiency that it can bring. It promotes a more efficient approach by preventing the conducting assessments again after a reliable authority has already gathered the same information. And another key aspect is when you use reliance, that you are still responsible for the decision. It does not mean that you must accept another authority's decision, but you can use that information to help you reach your own decision, taking into account your local context or the needs of the people who will be using or relying on your decision. So the two areas where we have introduced reliance into the pre-qualification pathway is in our abridged pre-qualification assessment and also in the use of the collaborative registration procedure. So I'll give you a little bit of information about both of those areas. Our abridged pre-qualification assessment was introduced to avoid duplication of effort and to also reduce the time that it takes for us to pre-qualify a product by focusing on the areas where WHO pre-qualification assessment brings added value. We know if a product has already been approved by a stringent regulatory authority, we have a level of assurance relating to that product's quality and performance in those countries where it's been approved. So we can apply an abridged pre-qualification assessment if the regulatory version of that product has already been stringently assessed and approved by one of the recognised SRAs, or if there is a regulatory version of the product that in itself has not been stringently assessed, but the manufacturer can provide us with evidence that there's no substantial difference between the regulatory version they intend to have pre-qualified. One of the big bonuses of this is it can reduce significantly the time taken to pre-qualify a product. So for an abridged assessment, we aim to have the decision reached within 100 days or 180 days. The difference will be on the performance evaluation component. We have two different options for performance evaluation. The shorter option is where the manufacturer organises the performance evaluation with one of the WHO approved laboratories. The longer option is if they leave it to WHO to coordinate that. And compared to the full assessment, you can see we're looking at either 270 days, if it's option one laboratory assessment, or 350 days. So we find we can significantly reduce the time taken to reach our pre-qualification decision when we are able to use the abridged assessment pathway. And on the screen there, I've included the regulatory authorities where we currently recognise those approvals as making the product eligible to go through the abridged assessment pathway. So just a little more information on what the abridged assessment pathway actually involves. Instead of providing a full product dossier, the manufacturer can provide a much shorter dossier, which will be reviewed by our subject matter experts. There will still be a manufacturing site inspection, but this will also usually be of shorter duration because many elements of the quality management system will not be part of that inspection. Instead, the focus will be on areas that are specifically relevant to use of the product in resource-limited settings, things like the risk management that's in place for the product, and also the way that stability has been assessed for in-use and transport. But there is reliance on an inspection audit report done by the stringent regulatory authority to address many of the elements of the quality management system. So that is also reduced in time. The performance evaluation does not change. So we follow the same protocol that we use for the full assessment. For that abridged product dossier, the elements that we ask the manufacturer to provide concern the way the product precision has been validated, stability studies, including shelf life, shipping, and in-use, the data they have for usability and human factors, and the labelling and instructions for use, as well as their risk management documentation. The other area I want to address in terms of reliance looks at what happens after a product has been pre-qualified, and this is the collaborative registration procedure. The idea behind this procedure is to accelerate country registration for products that have been pre-qualified through information sharing between WHO PQ team and national regulatory authorities. It is a voluntary program for the manufacturers of pre-qualified IVDs, and they need to guarantee that the same product that was pre-qualified will be provided to the country that uses the CRP for market registration. We need to make sure that we have a confidentiality agreement in place for the data shared, and we ask the regulatory authority to reach the decision within 90 days. As part of the CRP, we will share the dossier review report and any change requests, the site inspection report, and all of the data generated during the performance evaluation. At this point in time, we have 24 different countries that have agreed to use the CRP, although they're not all active at the moment. Some of them are still getting their processes in place to use it. But we have shared assessment reports for 20 different pre-qualified products with 7 different NRAs under the CRP. And we've found that it significantly reduces the time it takes for those NRAs to reach their marketing decision. I won't go into detail about what is in the reports that we share with them, but you will have these slides available to you so you can see they're quite comprehensive, and we hope that it gives the NRA all the information they need to help them make a decision about whether that product is appropriate for their market. So the procedure itself is an agreement between the manufacturer of the pre-qualified product, the WHO, and the NRA that is considering whether that product can come to their market. We need to make sure that we have the permission of the manufacturer to share those reports, and then we share them confidentially with the NRA. The NRA can come back to the PQ team and ask questions about any of the reports if they would like to do so. It's truly intended to be a collaborative process. And as I mentioned, we ask them to reach their decision within 90 days and let WHO know the outcome. In our team, we have had to do quite a bit of work with regard to our dossier review reports going back to 2010. These were originally intended only to be for internal use. So we have had to go back and make sure that they contain sufficient information to be useful to NRAs. We also have had to change the way we manage change request reports so that we can provide these in a meaningful way to any NRAs that are using the CRP for a product where a change has been approved. We also conduct CRP workshops and meetings with the NRA to give them more information about the program, and also make sure they understand the assessment process we conduct so they can make a decision on whether the assessment we do is relevant for what they do and if they can rely on that. We're all working at the moment to promote more awareness of the CRP. We've found it works quite well in the countries that are using it, but we have more work to do to make sure countries are aware of it, and also manufacturers are aware of where the CRP is operational. So we have a number of different strategies in place to help us achieve that. So I'll just wrap up by saying the benefits that we've found from incorporating reliance into PQ is that it can increase the trust in the PQ process with the NRAs. It has been helpful for us for optimizing resources, particularly using the abridged assessments. And it gives us a meaningful platform to collaborate with other regulatory agencies. It helps us make best use of agreed assessment principles, and we've found that it increases knowledge, not only in terms of being able to capacity build for NRAs that maybe don't have a lot of experience, but we also learn a lot about the member states that are using pre-qualification, and it can help us improve our processes. And most importantly, being efficient helps us with our mandate of facilitating access to quality assured IVDs. So thank you very much for your attention. I'll leave it there, and I'm happy to answer any questions that may come up. Thank you.
Muito obrigado, Suzy.
Thank you, Ms. Suzy Braniff. Your presentation really talks about several important issues when we're dealing with pre-qualification of medical devices, specifically IVDs. And that, well, by using all those tools and this possibility of collaboration, that promotes more agility to the process. One of your comments was actually regarding reducing the time for the conclusion of a process by almost 50%, which is fantastic. And that is something that we at Anvisa want to adopt, one of those reliance mechanisms. And maybe we are able to achieve that gain of time and access to market for products for patients. That is our main goal, to promote the access to safe and efficient products in the least amount of time. So that is a very interesting process. Thank you very much. I would have some questions for Ms. Braniff. And if we have any questions from the audience, then please feel free to send your questions using the QR code on your screen so that we can address those questions. So Suzy, first question would be regarding the implementation of the reliance mechanism at WHO in their pre-qualification program. How are the changes of products conducted? We know that the pre-qualification of a product is an initial process, but throughout the life cycle, products suffer changes. So how does WHO deal with that?
Yes, changes are certainly one of the more challenging aspects because for a lot of the products we've had listed for 10 years or more, there's been multiple changes that have taken place. So for us, even if a product was listed based on an abridged assessment, any subsequent changes would still need to be reported to WHO and assessed by WHO to see if they are accepted or not. So we still need to know about changes, even if the product was listed based on an abridged assessment. With regard to the collaborative registration procedure, NRAs that have used the CRP to list a product usually still ask the manufacturers to notify them of changes as well. But from WHO's side, if we approve any changes, we share the change assessment report with any NRAs that have used the CRP. And we've actually found this to be quite a useful process because in some cases, the NRAs that have used the CRP for a product have notified us that a manufacturer has let them know about a change, and we find that they have not let WHO know. So it gives us the opportunity to go to the manufacturer and find out why they haven't submitted their change to WHO. So it's providing us with a bit of added benefit that we have another point of information to find out what's happening with products post-listing. But it is an area that does require some resources to continually monitor the changes and make sure that our NRAs are notified, as well as our internal processes for approving the change.
Perfeito. Certamente as alterações, elas
Excellent. So I believe that all the changes are a challenge for any regulatory authority that would adopt that reliance mechanism. I would imagine that that is not different. I mean, that is not different for the WHO. One other question that I would have, Suzy, is that WHO, if I'm not mistaken, at 2021 published a document that dealt specifically with the reliance good practices for the regulation of medical devices. And there are 6 principles listed in that document that deal with the universality and independence of the decision-making of the agencies, transparency, respect to the legal basis of each jurisdiction or country, consistency and competency. So in our there are authorities that are already adopting these reliance strategies based on this promotion by WHO. I mean, are you aware of those countries, and could you give us some examples?
So I can give examples from my experience, which is the NRAs we work with the collaborative registration procedure. So we rolled out the CRP for IVDs in 2019, and we had 5 countries that originally agreed to participate. And what we found was, for 3 of the countries, they were able to implement the CRP within a 1-year period, but for 2 of the countries, they were not. And the main barrier we found was the legislation was creating a barrier to using reliance to reach a marketing authorization decision. And it can take some time for that to change. So we've found with bringing new countries into the CRP that we need them to make sure that they have the appropriate legislative environment in place to use a reliance mechanism. For the countries where that hasn't been a problem, the other barriers that can exist are a changeover in staff. Some NRAs have a relatively high staff changeover, particularly in the area of IVDs, which may only have a small staff associated with it. So that can create a slowdown in implementing and embedding a reliance process, because reliance is based on trust. So understandably, it takes time to build that trust. And if you are having a big changeover in your staffing, sometimes it doesn't really come to fruition. But we do have countries where this is working very well. So from the CRP in Tanzania, we have found that they've been quite successful with implementation, and for pre-qualified products, in some cases, they've reached their marketing authorization decision within 2 weeks, even though we're asking for 90 days. Another country that's doing really well with this is Rwanda. It's taken a little longer for them to get their system in place, but now they are having quite a good turnaround time when they are assessing pre-qualified products under the CRP, and we're seeing them reaching their decisions usually within 30 days. So we are getting some evidence that the CRP can be very successful in reducing the time to providing access to these quality assured products.
Perfeito. Bom, Suzy, muito
Excellent. Thank you so much, Suzy, for your presentation and for your remarks, and for also, sharing your experience with WHO.
Welcome.
Okay, so right now, we would like to move on to Daniel Yoon's presentation. So Daniel is one of the persons responsible at Health Canada for the implementation of the tool called eStar. This is a very interesting tool that has already been shared by Health Canada and the US FDA in some presentations in the IMDRF. This is very interesting, and at some point in the future, Anvisa will try to implement that as well. So Daniel, the floor is yours.
Thank you, Augusto, and good morning, everyone. Thank you for the opportunity to speak to you today. I'll be giving you a brief overview of Health Canada's use of eStar, as well as some background on how eStar relates to the work of IMDRF's Regulated Product Submission Working Group. So before I begin, though, I'd like to start off by giving a bit of background and context about Health Canada's current processes. As you know, we are the federal regulator of medical devices, and we are responsible for the sale, importation, and advertisement of medical devices in Canada. Medical device licenses are issued to the manufacturers of Class 2, 3, and 4 medical devices. The amount of data required with an application is proportional to the device's risk classification. Class 2s, since they are low to moderate risk, require an attestation that the manufacturer possesses safety and effectiveness data, but they are not required to submit it unless we request it. This leaves Class 3 and Class 4 applications as the ones we conduct scientific reviews on. Class 3 applications generally require a summary of the data, while Class 4 applications require the full data set. Although we encourage manufacturers to use the INDRF table of contents structure, it is not a requirement. We are also currently piloting the regulatory enrollment process, which will allow manufacturers to use web-based forms to facilitate submission intake. The pilot is expected to end in December, after which it will be implemented on a voluntary basis. So with that context, these two pathways illustrate how applications are currently sent to Health Canada and how we envision applicants will eventually be able to send their submissions. At the moment, we receive the vast majority of submissions through the top pathway, where a manually created TOC that contains PDFs is sent to Health Canada by email or some other form of media. However, with the eStar pilot, the TOC will be generated by eStar and will use PDFs, which will be sent to Health Canada. Eventually, we hope to use REP XMLs, which will allow us to use the Common Electronic Submissions Gateway hosted by the FDA. And that is represented by the lower pathway as well. So what exactly is eStar and why does it matter? eStar is a dynamic interactive PDF that is used to build submissions for medical devices. It was developed by the US FDA and contains features such as automation, guides, and verification in a single package that is easy to use. Because it has been programmed with IMDRF's table of contents, this allows it to build submissions with both harmonized and region-specific content. And you'll see on the right here, there's a screenshot of what the eStar tool looks like. And you'll notice that there are visual cues such as red, green, and gray bars to denote whether a particular section has been completed. And I've included a couple other screenshots to illustrate that in a bit more detail. You can see here that, that the application jurisdiction and the application purpose have been selected. And so they have green bars here to indicate that those sections are completed. And then incomplete sections are represented by red bars. And if there is a subsection that has not been completed, the overall indicator up top here will be shown to be red. And I'm hoping that you can see my cursor. On the right here is another screenshot. And it just shows you that you can attach documents to cover a particular section. And then there at the right, there's an area where help text can be displayed to sort of give additional information and context about that additional section. And based on user selection of the jurisdiction, the device class, and application type, eStar will populate related sections. Users will also be asked questions about their device's characteristics, and eStar will further toggle sections for additional questions. So since eStar is an FDA-developed tool, it needed to be tested to determine how it performs when used for other jurisdictions. The joint pilot between Health Canada and the FDA was launched in January of this year with 9 participants. We were encouraged to see a lot of interest in this pilot, and all 9 spots were quickly filled. Participants are required to submit their applications to both agencies within 6 months of being accepted into the pilot. The submissions in scope are new applications and significant change amendments for Class 3 and 4 devices. They do need to be submitted in English, and we are not accepting French submissions as part of this pilot. Other applications that are out of scope are combination products and IVDs. And then there are also some additional exclusions related to FDA-specific applications. But for the FDA, 510K, De Novo, and PMA applications are eligible for this joint pilot. And we are also conducting a Health Canada-only pilot to further assess how eStar performs. This pilot largely has the same eligibility conditions, and my understanding is that some manufacturers have chosen to participate in both pilots. Unfortunately, I don't have any results or updates on the 2 pilots I can share at this time. It's still relatively early in the process, but we are looking forward to the results and hearing about the experiences from everyone involved and sharing the results at a later date. So why is Health Canada choosing to use eStar? As I mentioned earlier, eStar allows for both harmonized and region-specific content by using the IMDRF table of contents. This should allow applicants to have a common set of information that can be submitted to multiple jurisdictions, while also allowing the manufacturer to tailor the application to a specific region. eStar is also user-friendly. Unlike the previous templates of folders that were provided, which required manually transferring PDF files into each folder of the TOC, eStar only presents the fields that require populating based on the selections made. For example, class and whether an application is new or an amendment. And then, of course, you saw from the screenshots that it's a relatively simple process to provide attachments. Similarly, eStar will only show relevant sections to users and remove unrelated sections or TOC folders from the submission. Removing irrelevant folders is a submission requirement, and without eStar, users currently have to manually remove unrelated folders. Once all the attachments are added and the submission is complete, eStar will convert the contents into the TOC format for regulated use. Because the conversion into TOC places the files into an XML structure, the technical limitations that were previously experienced, such as character length, are better mitigated with validation prompts. And then in part to geographical proximity, half of the manufacturers that hold Canadian licenses come from the United States. Health Canada has aimed to align with the FDA where possible, and we have worked with them to program our requirements into eStar. By using this common submission building tool and leveraging internationally harmonized set of common data, that could result in earlier submissions to Health Canada and facilitate earlier access to medical devices for Canadians. And so I'd like to spend the next few slides talking about the table of contents structure specifically, as eStar is now closely associated with it. The first edition of the table of contents was developed by the Regulated Product Submission Working Group in 2014 and used classification matrices, which were region-specific tables that informed applicants if a particular section was required, not required, conditionally required, recommended, or optional. And this table on the right gives you an idea of what the classification matrix looked like. The green rows represent required sections, while the unhighlighted rows represent not required sections. And then the last row is conditionally required and lists the condition in the right column. Templates were also created for each of the different folder trees. For example, for a Health Canada Class 4 new application, so that applicants could put a PDF in the appropriate folder or subfolder. A pilot was conducted in 2015 with a select number of regulators to determine how the TOC worked in multiple jurisdictions. Unfortunately, we found it challenging to find applications that could be simultaneously submitted to multiple jurisdictions. Feedback from industry was also mixed. Although some appreciated having a harmonized structure, there were also technical difficulties such as character limits that made file naming a challenge, especially when you got down into multiple subfolders The granularity of the TOC also meant that there were multiple levels of subfolders, so which made navigation a bit cumbersome. So taking this into account, IMDRF approved a new work item extension in 2021 that has two phases: updating the TOC structures for both non-IVDs and IVDs, and incorporating those updated structures into eStar. The updated TOCs were posted for public consultation earlier this year, and we received over 200 comments. The co-chairs performed the first assessment of those comments, and the working group will begin discussing them in the next little while. And this slide just shows the chapter divisions of the TOC. These chapter topics have stayed consistent over the years, and you can see that every chapter, except for chapter 3, deals with the same topic for both the non-IVD and IVD versions. And the next couple slides just goes over the minor and the major updates that we made to the TOC. I won't go into a lot of detail over the minor ones, but for the major changes, I wanted to flag that Korea and the UK joined the TOC. And so we've incorporated their regional content, although it was a bit limited at times. Post-market subchapters were added, and chapters 6A and 6B on quality management system were consolidated into a single chapter. And then there were also substantial updates for some regional content, specifically for Australia, Brazil, China, and the UK. And this represented regulatory, legislative, or policy updates. And then finally, this slide lists the membership of the RPS working group. In addition to regulators, the World Health Organization and EU notified bodies are also members. So what's next after the pilots? First, Health Canada and the FDA will review the results and analyze participant feedback, both from industry and regulators. In the short term, the RPS working group will analyze the comments from the recent consultation and make appropriate changes to both the non-IVD and IVD versions. The working group will also continue conversations with interested members on adding jurisdictions to eStar. Anvisa has been a great contributor to the working group, and as Augusto mentioned, they've expressed interest in adding the Brazilian requirements to eStar. In addition to programming Anvisa's requirements, the working group will have to figure out some logistical steps, such as adding Portuguese or other languages into eStar, and we'll be having those continuing discussions within the working group. And so with that, I'd like to thank you for your attention, and I will turn it back to Augusto.
Muito obrigado, Daniel.
Thank you very much, Daniel. That was a very clear and clarifying presentation on eStar. And indeed, this is a very interesting tool. We had the opportunity to learn more about it a few months ago. And as a regulatory authority, it brought ideas to Anvisa to even change our application flow here in the agency, because eStar is an offline tool. So it's not the petitioning process that we have at Anvisa today, in which companies need to prepare a number of documents and submit each and every one of them, which is quite a lot of work. And many times we have issues with the system. And eStar is an offline tool that allows the company or manufacturer to do all that work at a certain time and then just submit it to the regulatory authority in a second stage. So making this work much more convenient. Daniel, just one question for you before the next presentation is about the updates or the updates that have been made on the tool. Is there a plan on behalf of Health Canada and the FDA on how the updates will be conducted. For example, how to include new information fields or changes on specific fields that need to be improved or clarified. Has this been already studied by the developers during the pilot?
We do have ongoing conversations with the FDA on the updates and what needs to be addressed in the E-Star. It's not really a formalized process at this point, but it's just some ongoing conversations that we do have just to address some of the technical challenges or technical capabilities of E-Star. I imagine though, as the process becomes more formalized and we incorporate additional regulators into E-Star, for example, if Anvisa, there will need to be probably a more a formalized and regular way that we handle those updates and needs. But at this point, it's just through constant communication that we have with FDA to sort of address those.
Perfeito.
Perfect. And what about the use of the TOC? The TOC is a very interesting structure that was created inside the IMDRF, and Anvisa is already using it in our two main regulation areas for health products. So thinking about the TOC and how it is represented inside the different product regulations, certainly that is going to happen, or it will allow for E-Star to evolve. And I believe that most of the companies that work globally will be interested on using the TOC, of course, depending on the jurisdiction where they work, but they will also be interested in adopting the E-Star tool. So I think my question is, how was E-Star received inside Health Canada, or inside Canada, actually, by the industry?
So I'm sure Seema can speak to how E-Star was received on the industry side of things, but within Health Canada, we thought it was very useful. We liked how easy it was to use, and being able to provide a tool that industry would hopefully be happy with, especially in comparison to the current process where you kind of have to manually create those folder structures. And even though there are templates available, we did receive feedback from industry that it wasn't really the best way to sort of build a submission. So E-Star's a lot more user-friendly, and with the ability to convert the documents that you add into E-Star and having the tool itself convert everything into that table of contents format and structure. I think it just makes things a lot easier for everyone involved, and everyone's pretty positive about it.
Excelente. Bom, perfeito. Muito obrigado.
Excellent. Well, thank you very much, Daniel, on your presentation and for answering the questions. So right now, we have another guest, Ms. Seema Vias from Medtronics, one of the companies that has actually sent submissions both to FDA and Health Canada by using the E-Star tool. So I think Seema is in a privileged position to talk to us about the experience of using the tool and how the company or her company welcomed the use of the tool for the submission. So Seema, thank you very much for being with us. And please, the floor is yours.
Sima, estamos sem o seu áudio.
Sima, we cannot hear you right now. Just a second, please.
Can you?
Yes. So now I can hear you.
Sorry, can you hear me now?
Yes, now we can. Thank you. Perfect.
Yeah, I think it was from my end. I was trying to unmute, but couldn't successfully do so. So thank you very much, Dr. Augusto, for the introduction. And I put together some Portuguese. So bom dia. É um prazer estar aqui. I hope I'm justified those two statements in Portuguese that was provided by my team. So first of all, thank you to Brady and Anvisa, specifically Dr. Augusto and Dr. Sergio for the opportunity to present our experience on the electronic submission template and resource via the participation that we had opportunity to participate in the pilots. And I think hopefully this presentation may also address Dr. Augusto's question on how the industry has perceived this eStar tool and our experience. So before we go into eStar, I'm very thankful Daniel set the stage of what eStar is about and walked you through some of the background information on eStar. I would like to take this opportunity to walk you through some of the information and some of the background that happened and where Medtronic collaborated with the regulators to have eStar where it is now today. So walking through the eStar timeline, as you can see, in February 2020, FDA launched the eStar pilot, and they announced that pilot in 2020. In September 2020, FDA encouraged applicants to begin voluntarily using eStar. And approximately in another 6 months, April 2021, Medtronic, started working with FDA and Health Canada in the background to beta test the eStar tool with IMDRF TOC structure. Now, the FDA regulator who designed and who's the brain behind eStar tool, while he was designing the tool to incorporate and make it user-friendly for the industry and regulators, and he incorporated the FDA requirements, but he also, in keeping in mind the environment that we are in, he also incorporated the IMDRF TOC content in the same eStar template. And that really helped us to help with and work in the background with FDA and Health Canada so we can beta test and make that ready for the pilot that will be announced. And in May 2021, as you can see, FDA noted it is going to see if eStar can be leveraged by IMDRF. And the main purpose that they said was to try and get a more standardized format for pre-market submission. Earlier this year, in January 2023, both FDA and Health Canada announced the eStar pilots for certain devices that Daniel just provided and presented. And the pilots were 2 pilots that were announced in parallel. One was specifically for Health Canada, and the other pilot was a joint FDA and Health Canada pilot. And I'll show you a little bit more into my presentation on these pilots. Looking into the future, it seems eStar expansion and international pilots with other regions after Health Canada might be a potential and something that we can move forward with. So with Medtronic, we were accepted into the pilots, and we were accepted into Health Canada only pilot, as well as the FDA and Health Canada joint pilot. For the Health Canada only pilot, we had the opportunity to get selected for submitting the Class 4 license application for a change, which is related to sterilization. And we also had the opportunity to submit and experience the Class 3 new license application. For the FDA Health Canada joint pilot, the submission that was selected went through the FDA traditional 510(k) pathway. And for Canada, it went through a Class 3 license amendment line extension. So in the next couple of slides, I'll try to provide you some glance into eStar. Daniel showed you from a regulator perspective what eStar is and provided some brief. I'll try to walk you through a little bit more granular detail and try not to repeat what he just said and presented and make it more objective for you. So this is a screenshot of the application submission type section. And it gives the opportunity for the user who's using to build their submission to select between the applicable jurisdiction, whether it's US FDA or the Health Canada, and potentially others can be added here in the future. It also gives the application purpose, which is by selecting the specific classification. And as we go down here, I hope you can see my cursor. You can also have you can also choose the application type. So once we select these information, as you can see over here, that Daniel was kind of pointing out is the green bar. And once this becomes green, it means you have completed the requirements of this section and it is good to go. There is no deficiency on this particular section. So that's how it's very helpful, as it prompts the user on the questions that is applicable to the specific jurisdiction, the application purpose, and to the type and change of the application. Moving a little bit more into the application, you can see that when we come at the cover letter, the way to build this submission is by attaching the relevant files. So you can start attaching, have your cover letter prepared, and all you have to do is attach it. Now, if you want to understand some of the requirements and you want to know if you are fulfilling the requirements, all you have to do is start clicking on the question mark. Once you click on the question mark, it will provide you with the guidance specific to the jurisdiction and the type of section and the application you have selected. And as Daniel mentioned, this is all IMDRF TOC guidance since we were choosing to work on the Health Canada submission. Same thing for application form. You attach your medical device license application form. And once, as you can see right now, this particular section is red, and this is how it looks. But once you attach a cover letter, attach any letters of reference, and attach all the information that is prompted by the template itself, this red bar will turn into green. So it's very, very user-friendly, and it's very, very helpful because you know that now you have completed this section and you have provided all information related to your application required by the section. This is a little bit more granular. As I was mentioning, it just provides you a device description section that is a little bit more detailed. And as you go into the submission, it will prompt you with specific questions. So in this particular section, depending on what we choose, what the user choose related to the device class and the application type, it will prompt all these questions. And once you start addressing and answering them, then it will start building and giving you the prompt and start giving you the green bar. As you click on the question mark, this is what there would be a dialog box like this that will open up and will prompt you with the requirements and what the recommendation is. So as you can see, the good thing about this and what is very useful we found is you don't have to open another guidance document or look into another document. Everything is built within the eStar template itself. it's just a matter of a click. You click the question mark and you get the information and the guidance to help you further your submission. So as you add attachments for the guidance, instructions are provided, questions are asked, and all you have to do is just follow those instructions, follow the guidance, and start attaching your data, your requirements as an attachments. All you have to do is also ensure, keep an eye on the bar, make sure it's always green so that you have, that indicates you have completed and have all information. At the end of your submission, a verification toolbox, pop-up, a verification section comes up like what I have on the screen, and it really indicates the following sections are complete. So all the sections that were completed and required by your submission will be listed here. And once it has this green bar, it indicates all your sections are completed. If you have a red bar, that means it will prompt you to go back to that section and fill in the gap, address it right while you are putting and compiling the submission. This is a slide I thought would be very helpful, and this is because we had the opportunity to also participate in the FDA and Health Canada joint submission. So what happens is, in the joint submission, the user have the ability to choose or select the specific jurisdiction they are applying to. If they are choosing to prepare the US FDA submission first, then once they choose the US FDA submission, they will start building the application. Now, the way we worked is, once the FDA submission was prepared, the same file was shared with the Canadian team, and the Canadian team, when they chose and selected the Health Canada jurisdiction, this particular notification comes up, which indicates that by switching jurisdiction, some of the attachments that are no longer required for Health Canada would be removed. And the good thing and the beauty about this template was all the related attachments that were common between the submissions selected for FDA and Health Canada. They were all transferred into the Health Canada format. And that really is helpful, as it does reduce the submission time and the duplicacy of efforts that we most of the time have to put together when working on submissions for different jurisdictions. So I heard Susie mentioning earlier in her presentation about reliance, and also some of the principles is avoid duplication of effort, reduce time to prepare submission. And this is just one great example right in front of us that helps us to meet that particular principle. So once the FDA eStar template is completed, we convert it, we start putting the attachments together, and the attachments were transferred for Health Canada, and it became easy for us to compile our submission for Health Canada. And this is just showing the options of how to select the jurisdiction, and the green bar meaning sections are completed. So based on our experience, I'd like to share what worked for us, what didn't work, and what are some of the opportunities for the future. So basically what worked is creating the submission in eStar was user-friendly and very easy. That's definitely something we all found while working on these pilot submissions. The other great thing about it is, although the eStar uses IMDRF template, there isn't a need to create the granular folder structure and worry about the character limitation, as the manufacturers who participated in the IMDRF pilot, and I'm pretty sure Brazil was one of them, can understand the pain we went through when trying to put the character limitation together and make sure all the granular folders are being appropriately captured. Instructions throughout the template we found, as I mentioned, are very helpful. And the good thing about it is, if a manufacturer is using a publishing tool or there's a need to publish. By using this template, you're building the submission in the template itself. So and that's what you would be submitting. So there's no need to publish. So you're eliminating a big step into the submission preparation process. It also building submissions by attaching your PDF file. So you can work offline, you can put your Word documents together for all the sections, convert them into PDF, and just start building those and adding those PDF in eStar. What didn't work is put together from a Health Canada perspective. And for us in Canada, we do have the opportunity, as Daniel was mentioning, we can either send a CD or we can also email. Now, when we are using email, because the eStar template is one big PDF file, we cannot break it into different and split the big PDF file. And most of the time, the submission will be greater than 1020 MB, which cannot be emailed. So that definitely is a limitation. Sections not applicable do require an attachment or it stays red. So you do have to keep on adding a rationale why it's not applicable. And that, I think, is something that can be improved. Some of the field do have a character limitation, and authoring submission, for example, inability to capture the track changes on the eStar template itself is also a limitation and didn't quite work. Now, I would also like to say these are some very high level feedback that definitely are opportunities that can be, these are things that didn't work, but they're not roadblockers. I believe these are things that can be addressed once we give the feedback to the regulator, they can be incorporated. Looking into opportunities for future, there is option to submit via the common electronic submissions gateway. And I think Daniel already spoke about it, and it seems they are already working towards this. Option for selecting the change type and applicable sections, similar to the US template, which we found the US template is a little bit more detailed, and maybe there's opportunity that Health Canada can also have additional fields that can further reduce or eliminate some of the requirements. We talked about harmonized requirements or utilized reliance. I think that is something that would be very helpful for eStar and moving forward with other jurisdictions. And I'd like to share with you one of the key area where we really were challenged when working on this particular eStar submission. And it's more not related to the template, but it will be more related to the regulatory environment that we are in and the regulatory landscape of US and Health Canada. For example, the US was using 510K, and the premise is substantial equivalence using predicate devices, whereas Health Canada does not use substantial equivalence. They do recognize the predicate or the previous generation or other competitors' product. However, the challenge for us with this was if the predicate that is used in the US submission is not licensed in Canada, then the whole point of leveraging the same documents, the same attachments using eStar does not hold. Because now we ended up, what we had to do is we had to remove those documents and make it specific to the Health Canada requirements. So it's nothing specific to the eStar template itself. It will be the same situation if we had to do the submission outside of eStar. But my point here being is, these are some of the areas where harmonization or reliance can really help and help us achieve the goal. Also, ability to work on some files while creating submissions for different jurisdictions would be something that would be very ideal. An expansion and international pilot with other jurisdiction geographies after Health Canada is something we're looking forward to. There's also opportunity for hopefully a single dossier or a single review in the future. So overall, the eStar benefits that we look at and see from a regulator's perspective or the industry perspective gives us 4 main themes. And from a regulators, we see there's efficiency and global access. And from industry perspective, we see it's global harmonization and it's the ease of use. So from a efficiency perspective, if we just focus on that, how it brings efficiency is it helps regulators review the application more efficiently as the information is standardized. And the efficiency is built both in terms of time as well as resources. And there's potential to leverage the gateway access in bringing more efficiency into those jurisdictions like Health Canada that currently do not have the ability for eStar. So there is where the efficiency piece lies. From a global access, eStar definitely provides a platform that regulators to improve the regulatory landscape and further the mission of accelerating the global access. It's a step towards a single review process, an opportunity to enhance the collaboration between regulators and will help them understand each other's regulatory landscape a bit better. It also potential for increased collaboration between industry and regulators, such as this pilot project. From an industry perspective, it's a step towards another step towards global harmonization, where in addition to US FDA submission structure, this template incorporates IMDRF, and it facilitates the global harmonization through a single dossier in the future. There's potential to reduce regulatory burden and potential to expand to other countries. The ease of use, as I was demonstrating, walking you through the eStar template, it's very user-friendly, and it guides the user while building the submission in the template. And it avoids any gaps or potential for additional clarification questions from the regulator during the screening process. Uses a more accessible platform that is enabled for mobile use and allows sharing an eStar file with team members while it is being prepared. That would be something that we are truly looking forward to. So from an industry experience perspective, our overall experience has been, it has been a very good experience. And we highly recommend moving forward and look forward to collaborating with the regulators to pursue eStar and have this as one of the options to build our submissions and submit to the regulators. So I know we'll have Q&A, so would be happy to take any questions that you may have, and would like to thank you for the opportunity and the time. Thank you so much.
Muito obrigado, Sima. É muito interessante a gente poder ter essa oportunidade de conhecer a experiência de uma empresa.
Thank you so much, Sima. It's interesting to hear the experience of a company that has been using that type of tool. These tools are really important for us so that we can improve the convergence that we are looking for, the regulatory convergence. This is something that IMDRF and all the agencies and jurisdictions that are part of IMDRF are really putting a lot of effort and dedication. And so I really believe that a tool like eStar is really a leverage for us to meet those goals faster. And the goal ultimately is the benefit for all users of medical devices, for all the patients that, you know, and the population that needs and seeks efficient healthcare. Unfortunately, we do not have any more time for a Q&A session, but I would like to ask our panelists to answer some of the questions via email if they are available to do so, if that is adequate. So that would be very nice if you could do that. And we would later on share your answers with our audience, both online and here in person. Finally, I would like to thank Susie, Daniel, and Seema for being here. It was an honor to have you here at the seminar, and we hope to meet you again in the future, and that we are also able to move forward and progress in the area of regulatory convergence, reliance, and harmonization mechanisms. Thank you so much, and have an excellent day.
Thank you so much.
Thank you.
Have a good day ahead. Bye.
Vamos lá? Sim. Em nome dos organizadores.
So, on behalf of the organizer, I would like to say thank you to Ms. Susie Braniff, Mr. Daniel Yoon, and Ms. Seema Vias for their presentation. I'm going to ask Mr. Augusto Gaio to remain on stage. Before two quick messages, I just want to say thank you to Angela Marcos from ABIMEV, Mr. Leitão from SIND-OSP, and Mr. Geoffrey Moraes from ABIMO. I also like to inform you all that together with an evaluation survey, you will receive all the presentations electronically, all the presentations that happen throughout the 2 days of our seminar. Now, I would like to invite Ms. Cassie Scherer, Senior Director of U.S. Regulatory and Global Digital Health Policy from Medtronic USA, to talk about the use of apps, electronic systems, AI for the creation of application dossiers for medical devices. Welcome, Ms. Scherer. So while Cassie sets her computer up, I would like to apologize that we were not able to update the CVs from the different presenters to the platform. But I would like to ensure to all of you that their CVs are unquestionable. When we see someone taking the stage so casually, we have no idea about the importance of their career. So I'd like to invite you all to learn more about each of our speakers later on. Cassie is not only responsible for the U.S. regulatory and global digital health policy, so working with softwares and different intelligence mechanisms, she has also worked at FDA on these and other issues related to the regulation of these elements.
Hello, hola. Thank you so much for having me here, and I feel so privileged to be here, also in this wonderful country of Brazil. All of you are so welcoming, so obrigada to all. I'm Cassie Scherer with Medtronic, Senior Director of Global Digital Health Policy. Before Medtronic, I was also a regulator at US FDA. I'm going to talk today about something exciting and something I am very passionate about, really the opportunities of artificial intelligence, AI, and the benefits to healthcare and medical devices. Also, really, for those of us in this area, the rapid evolving nature of regulation of AI. If you want a career, this is it, because there is a lot going on in AI right now. At Medtronic, we really see the tremendous potential that AI-enabled technologies can bring to patients and physicians, but only if done responsibly. We at Medtronic put out an AI compass externally that really affirms our commitments to put out AI ethically and responsibly. So I'm going to talk today about some of the opportunities of AI-enabled medical devices. There's so much potential benefit that they can bring to patients and physicians, as well as some of the current regulatory challenges, and then touch on the global developments that are happening in addition to the evolving regulatory frameworks. And I was so pleased to hear some of the great discussions around harmonization reliance, because I will also touch on that in my presentation as well. So here's the agenda. It will be a lot of information, but hopefully I broke it down in a way that is understandable. So first, I just want to go into what is AI. Often we talk about AI, and as those that live in this space know that there's lots of different definitions around AI. Then I'll provide an overview of AI and medical devices and discuss some of the ways that the regulatory frameworks are evolving and what's expected for the future, in addition to cover some unique considerations around AI. What is AI? This is a really hard question. There are a lot of definitions out there. And if you are in this space, you know that the EU has put out a draft legislation in this. Australia and UK has also put out some proposals for consultation. There's all different types of definitions out there. And so really breaking it down, I put some of the terminology on here is from ISO standards, really again, trying to look to standards to be harmonized. AI, artificial intelligence, is really a broad term. And really what it boils down to is the act of a computer simulating the intelligence of a human for a particular task. What is AI can change over time. It's really in the eye of the beholder of what it means to be human intelligence. For example, we've dubbed image classifications as AI so that determine whether it's a dog or a cat on the screen. But in the future, you could maybe see that wouldn't be thought of as AI. Going down a layer deeper is machine learning. And we'll go a little bit more in the next slide of getting into an example of this, but really what it is as opposed to a scientist building an algorithm, it's really letting the machine do it itself. So if you were trying to figure out whether or not an image is a cat, you would feed the computer some input data, and we'll go into an example of this, and then feed it a target or task, trying to determine whether the image is a cat. And then you determine the type of the architecture of the algorithm. And then the machine is what optimizes the parameters of the particular architecture. So looking at the image and determining whether or not, if it's a certain color, that it potentially could be a cat. In machine learning, you also have deep learning. So that's a subclass of the machine learning model. And this is where we've really seen the breakthroughs. Deep learning is associated with a neural network that is loosely emulated off of a synapses of a human brain, but fairly loosely based on this. And this is really where we're seeing a lot of the performance. And people sometimes say deep learning and AI interchangeably, but it's really not. AI is an umbrella term, and deep learning is a subset of that. I also put on generative AI, which is ChatGPT, because these days everyone wants to talk about ChatGPT. I will not be touching on this in the presentation, but I do want to talk about what it is. So again, a subset of machine learning systems that are trained on a vast amount of information and then able to generate predictive responses. And for those of you that have used ChatGPT or other generative AI, you know that these responses can be in the form of text, images, computer code that simulate human responses. So just going a little bit more into, I know that's a lot of information of what I mean by this. So just to break it down, traditional algorithms, as we know, have been around for last 100 years. Machine learning last 30, 40 years, and deep learning for the last 10 years. So if you look at a traditional algorithm, that's the top of this slide. If you get an image and want to classify whether, oops, hold on. I forgot. There we go. There's our car. If you get an image, so you want to determine whether or not a car is from an image, the approach would be to have that image. And as an engineer, you would extract features from that image. So for example, you would determine the intensity of the color or the pixel parameters, And if those pixel parameters met a threshold, you would build a classification model manually. So for example, if the color intensity or intensity parameter is greater than X, it's a car. So then comes machine learning around 2000. Once again, as the scientist, you take the images, you extract the features, intensity of color, but instead of the engineer building a series of if-else statements, so if a certain parameter, it's a car, if not, it's not a car, you feed the features plus the data with labels to a machine, and it automatically creates those if-else statements. And so we saw some performance boost on this. And then around 2015, I started to build deep learning models and create these end-to-end models. And then what it is you don't extract those features, but you feed the images directly to the neural network, and it will extract the features, the intensity of colors, and build a classification simultaneously. So as you can imagine here, you're taking away a lot of the work on the front end, and you see a real performance boost. We have talked a lot about feeding data. And the takeaway from this slide is that really, and this is really important throughout my entire presentation, that large quantities of high quality labeled data is key to AI success. And I will build that thread throughout this presentation. But if you look at the chart on your left, you'll see that the success of AI is fueled by the size of the data. So if you have less data, the traditional algorithm will outperform the AI. But as you get more data and high quality data, the AI starts to outperform the traditional algorithm. Also, you'll see that the level of difficulty building an AI model is correspondence with the amount of labeled data. Okay, so we're past the sort of setting the stage, and now we're getting to the exciting part of what does this mean for medical devices? Hold on, I got so excited, I flipped through all my slides. Okay, all right. So why are we so excited about AI? And now we're bringing it down to healthcare and the benefits for physicians and patients. As we all know, with our watches, our connected watches, our wearables, electronic health records, there is a lot of data out there right now. And this increased amount of data offers the ability to see patients in their home environment, to better understand the patients and their conditions and how they're operating. But it is an explosive amount of data that right now is really hard to get any meaningful information around. I know, speaking to physicians, they'll tell stories about patients coming into them and saying, well, my Apple Watch said this, what does that mean? We need a way to really get all this information to a way that's meaningful and curated. And this is what AI provides opportunity for. It really provides this opportunity to analyze and curate vast amounts of data and provides meaningful output. But again, importantly, which I'll talk about, it needs to be leveraged responsibly. And we need to make sure that the regulation around it allows this innovation. And also, another potential too is really gain a more holistic opportunity to look at patients from the whole continuum of care, to have the patients more connected to their physicians. The other thing I wanted to touch on briefly is some of the future cases of AI. We talk about the benefits of AI to medical devices, but you can imagine benefits of AI across the total product lifecycle of the medical device. From the design stage, starting to look at generating design prototypes for medical devices, to clinical trials to help with patient recruitment by identifying and screening potential participants based on inclusion and exclusion criteria, to manufacturing, such as in predictive maintenance. Also, you think about regulatory submissions and putting them together. Again, AI can be used to extract and analyze large, vast amounts of data. So you think about clinical trial results and adverse events reports, being able to use AI in that area. We'll talk a lot about iteration of products once on the market. And then last, I'll just mention post-market. With complaint handling, for those of you in the space know that complaint handling can be quite intense and complex. AI can also provide the ability to streamline and improve complaint handling processes. For example, earlier detection of issues or trending of issues. We talk about a lot about AI like it's new. The point of this slide, and this was taken from an analysis of US FDA data, is that AI is really not new. We're just seeing more of an explosion of it in the last few years. And really the explosion of it, again, my theme comes with this more vast amount of data and high quality data. Like you'll see on this slide, most of the experience has been in radiology. And, for example, image analysis tools, really assistive tools to help the radiologist with detection of a disease or triage for the radiologist. And again, the reason for this, you guessed it, is because there was a large amount of readily available data in this space that were used to help develop and train products to interpret CT and MRI and plain radiograph. We're also starting to see, though, and you'll see as you go towards the right here, an explosion in other areas. In cardiology, Medtronic has products in this space, as well as GI and surgical. And we're continuing to see more of a diversity in AI with different disease states, as well as we get more access to data. So now we'll get into the regulatory framework. The current, as regulators across the globe are looking to see what changes need to be made to the current regulatory frameworks. As a baseline, the current regulatory frameworks were really meant to be for all medical devices and really provide a robust mechanism for ensuring the safety and effectiveness of all medical devices, including AI. And sort of the takeaway message is we don't need to create something new because we really have this robust framework already in place with medical device regulation. As we know already in this space, the medical device framework regulates sort of the pre-market aspect and as well as post-market and looks at the intended use and risk. And AI-enabled devices are really no different. What we'll talk about, though, is some unique aspects of AI where there is a need for some of these frameworks to evolve in order to encourage the innovation. So what is unique? We talk a lot about evolving these regulatory frameworks and what's unique about software, including AI. One of the biggest things, and I'll talk a lot about this, is as we know, the regulatory frameworks are based on our traditional devices. So we think about hardware, pacemakers, products that take a long time to evolve and come back for a new submission to Anvisa and other regulators. Then we think about AI. The benefit of AI is that as we get more data, we can evolve it and we can improve the AI. What I'll talk about a little bit more is the need to evolve our current regulatory framework so that the AI can continue to evolve without having to go back to the regulator each time. And I'll talk about a unique tool that was put out by US FDA on the predetermined change control plan. The other thing I'll note is that it's really, in this space, important to have the right subject matter experts in this area that really know the technical space. We at Medtronic have done submissions on this and seen the benefit of our technical scientists being able to talk to the technical scientists at the regulator. I'm a lawyer by training. I've learned this space, but it's my R&D folks that really know the technical area and can talk about the models in a way that I could never talk about. With that, it's really important to talk about capacity building and enhancing regulatory efficiency. And I was so pleased to hear about the discussion on regulatory reliance, because this is really one of those spaces where it can help regulatory reliance can help make best use of the available resources and expertise while still ensuring the regulator independence and authority over the marketing of device. So here's some of what the next few slides that I'm going to talk a little bit more about is really these, where do we need to enhance our regulatory framework? Again, we don't need to start over fresh, but we need to think critically about where in our regulatory framework today we may need to evolve for AI. First is avoiding unwarranted bias or unintended bias. As we talk about this, and I'll talk about this further, as we all know in the healthcare area, bias is not unique to AI, but instead that there's important considerations when developing the AI model, not to further bias that's already in place. A big thing that you'll hear me talking about is enabling this more rapid iteration. Again, as I talked about, the frameworks, traditional frameworks are really built for more of these models that, or more of these devices that modify at a slower pace. And so thinking about tools like predetermined change control plan, which I will get to next, that are innovative ways to help with more rapid iteration. And then the other big thing I'll talk about is, I just want to mention, is labeling and transparency. This is something that, as we talk about building trust in patients and physicians, is so important. It's important as we rapidly iterate to be able to keep up with the modifications to labeling. Again, we have in the medical device framework, a labeling framework already in place, not something that we need to start anew, but to think about how do we enable this rapid iteration. One of the things that is a very effective tool is electronic labeling, electronic instructions for use, which I know in Brazil with Anvisa has some allowance for EIFU, but it is so key to this space because as you think about the rapid iteration, the ability to put out, almost automatically this labeling to physicians and patients, and also the allowance to make it more dynamic, to make sure that your patients and physicians can understand it. All right, so now we're going to move into something I'm very passionate about. So I'll try to keep it short, but it's change management and predetermined change control plan. Whoops, go back one. I've already really covered this of the opportunity with AI of this more rapid iteration, but with it comes a need to look at our regulatory framework, both from the regulator's point of view, so that they don't have to look at multiple submissions as different modifications come, and also from innovation point of view to ensure that we're really giving AI the opportunity to do what it does best, to be able to modify it once it's in the marketplace to improve and enhance it. So one of the tools that US FDA had put out there is the predetermined change control plan. I'll note that UK MHRA has already signaled that it will be starting to utilize predetermined change control plans about it as well. So what is it? I know I've mentioned its name a bit, but really what it is that it's something that when you go to the regulator with your pre-market submission, you go with your pre-market submission and you also go with your predetermined change control plan. The predetermined change control plan, or PCCP, allows you to put into your submission specific post-market modifications that you plan to make. And provide that with your pre-market submission. You provide the regulator what your specific modifications would be and also how you plan to do it. Then when you're approved by the regulator, you can get on the market and make your second generation, third generation modifications without having to go back to the regulator. It basically puts all that post-market work into the pre-market setting. And it's a lot of work in the pre-market setting, but it allows for this faster innovation once you're on the market. Again, allowing for this innovation to happen and also bringing efficiencies both to the regulator and to industry. We've been talking a lot about this globally as well with different global regulators. So I really appreciate the opportunity to speak about this with all of you today. Now moving on to AI and bias, and I'll just move through this. Quickly, I know I'm probably almost at my time, but bias is one of the hottest topics in AI. You hear it mentioned so much. I put on the definition on here, again, the definition from ISO, that's also leveraged by IMDRF of what bias is: a systematic difference in the treatment of certain objects, peoples, or groups in comparison to others. However, we'll see a large amount of different definitions of bias. Out of the healthcare space, a classic example is the facial recognition software. I'm sure a lot of us have heard about this in the news at some point, where a facial recognition software was developed using data from mostly white faces with not much pigmentation. Then when AI was put out there and exposed the model to faces with more pigmentation, it produced results that didn't work well. It's because it was never trained to understand the differentiation between different pigmentations in skin. Well, the same thing can happen in medicine. Again, going back to this initial comment about the need for large, high quality data. If you don't feed it the right if you don't feed it the right data, it's going to produce outputs that are not correct or not high performing. The causes of bias can be many, but it's not limited to AI-enabled products. On this slide, sort of the main takeaways that I do want to just point out is that we talk about bias being negative, but it can also be positive. We are aware, I gave an example from not in medicine, but another example of bias, unintended bias, is when you train it using systematic bias data. So for example, if we had, as we know that women can show symptoms differently than men in cardiac disease, if we're therefore training our AI systems on electronic health record data just from men, it could potentially misdiagnose myocardial infarction in women. So again, making sure that we're using diverse representative data. But I also want to mention that bias always isn't bad. Sometimes we are intending to create bias. And what do I mean by this? Sometimes we develop tools that are optimized for a specific population. So when it is known through clinical studies that certain subpopulation express symptoms differently than other populations, we might optimize our tool, that product, for that intended specific population. In that case, yes, it is biased to that population, but it is for good because we're trying to make sure that this device is performing for that population. It's an important thing because as we think about bias, we always think about negative, but it's important to recognize that in some cases it's intended. It just needs to be make sure that we're labeled for that population. I have on here trying to make it a little bit more fun, Stonehenge, because as we know, bias is not new to AI. It's as old as humans. We as humans learn by patterns and assumptions, and we are flawed. We have bias in us. In the medical care area, the same thing. There's a long history of bias in healthcare. And also in the medical device, and you think about just sort of something simple as developing sizes for medical devices that sometimes you don't take into the account the needs for the entire population. The last thing I'll note on this is that as we are looking to mitigate unwarranted bias, and as we're looking to make sure that we have appropriate representation of our data, as we determine what is appropriate representation, really the important thing is understanding those variables that can impact performance. So for some medical devices, things like skin pigmentation, race wouldn't matter. But instead, there could be something like BMI or imaging inputs that would matter to the performance. So we need to make sure that we're really taking into consideration what's the intended population and the expected use, and then also what is the variables that could impact performance when we're developing our AI. And so, going along this thread of needing really representation, data is very much like food to the AI systems. We know we are what we eat. So, and same with AI. It is what how good an AI system depends on how good the data is. Data sets need to be representative of the intended population. As we know, data privacy laws attempt to protect sensitive data and similar variables. And this can be in tension with a need for this data in the development of AI to ensure that we have these accurate and unbiased results. So one of the points of this slide is really to make sure that we recognize that sometimes there is this tension, that sometimes we need certain variables in the patient data in order to train the AI so that it's unbiased. If you restrict access to features like race or gender or disease states or other things like BMI, it can become difficult or even impossible to determine if the AI model is biased and whether or not it's performing accurately. And then the last slides I'll just touch on really quickly is harmonization. I've already touched a bit on the fact that it is so important in this space to have harmonization and also use tools like Reliance. We are seeing regulators across the globe put out different proposals in this space. And it's really important that we're using definitions and concepts that we can all agree upon so that we avoid confusion and inconsistency. I also know, again, we have these regulatory frameworks in place. As we look to develop AI legislation, a lot of countries are looking to their regulators, like Anvisa, to say that they already know how to regulate medical devices, including AI-enabled medical devices. A couple of things I'll just note really quickly is these good machine learning practice principles that were put out by Health Canada, UK MHRA, and FDA. And really, these principles were meant to be put out as the beginning of a conversation to inform further work by IMDRF. They're not policy, but really meant to put out sort of what are the key areas of oversight focus that we should be looking at. And you'll see on this some of the same themes that I've been talking about, sort of the appropriate and representative data sets, training and testing considerations of the data, and bias and transparency. And then finally, I'll just leave you with sort of, again, with a plug for IMDRF. When we talk about harmonization, IMDRF is such an important place for a lot of this work. They have already put out a lot of documents around SAMD, software as a medical device, definitions, what is sort of thinking about what is a medical device, software as a medical device, and then also how do I classify, what's the risk classification, what are some of the clinical considerations and quality. There's also an AI definitions document. And the AI ML working group in IMDRF is being stood up and is going to start doing more work in this area. I understand maybe it'll be looking at the good machine learning practice principles that was on my last slide. Also, the CMD working group that I'm an industry representative on is also doing some work to put out some documentation. This is really important work for the regulators to get to, and with industry to all come together and get consistent viewpoints and interpretation. So with that, just leave you and thank you so much for this opportunity.
Bom, muito obrigado, Cassie, sua apresentação.
Thank you so much, Cassie. Your presentation was really, really interesting and almost provoking because we have several areas that started using machine learning and AI, and that has been increasingly demanded in every other sector of the society. This is something that we see that has, taken over the news, and we are daily hearing things and hearing new things and learn new things about artificial intelligence. And sometimes that gets scary, especially because of the speed of how everything's happening. In that sense, I would like to ask you, how do you see the possibility of regulatory agencies using these mechanisms of artificial intelligence, not only for us to receive information produced by the industry, but also to be able to assess and evaluate these products with the perspective of a regulator? Do you see that as something possible? Is there any room for agencies to evolve, to use these tools to make the assessment of a medical device process more agile, for example, so that this assessment is kind of deeper or less detailed? Or, do you see that, is there any perspective in that sense?
It's a great question, and I think the answer is yes. But it's still sort of developing. I could see, again, AI offers the ability to extract large amounts of data and analyze it in a way that's meaningful. And I could see a regulatory agency being able to better, on your own end, sort of look across submissions and see any tracking or trending areas. I also see the ability, for you to be able to sort of utilize, ensure compliance with submissions and guidances or other documents that you have out there. So if you can analyze to see whether or not it's meeting certain standards or regulations and things like that. We are seeing an explosion as well away from the submissions, just in internal company tools as well, to increase our ability to do effective work. So I could see even away from submissions itself, the regulators being able to do that. So the answer is yes. And I think it's just, again, ensuring that we have the right models in place to be able to do some of this analysis.
Perfeito. E nesse mesmo...
Perfect. And considering the same scenario, medical devices and the wearables, are we able to get information generated by AI in order to allow us to develop new applications or new indications of use of medical devices, to better use the information generated by medical devices, whether it be imaging diagnosis or vital signs that we might get from smartwatches and other devices? If this information pieces are put together, do you think they might allow the creation of new indications of use for devices that already exist on the market?
Yes, I think, again, one of the benefits of AI is that we will be able to start combining the wearables with other medical devices to create sort of a more personalized diagnosis, for example. And that combination will create a new medical device, right? Because it will have a different intended use. Again, some of this gets down to the ability for interoperability for data to be able to work together and building that model in a way that works and makes sense with the good data. But I do think that we'll get to a point where we can combine, the wearables with other types of medical devices. But again, I do see that as a new medical device.
Bom, eu não sei se nós temos
Well, I don't know if we have questions from the audience. Anyone would like to ask Cassie a question, please feel free to do so. I'm sorry, questions need to be asked on the mic so you can hear them. Well, another thing that is often brought up, not only in the agency, but from different sectors, has to do with data security. Data that is being shared by companies and also health services. So how is that being dealt with, considering the rapid evolution of these mechanisms and the AI applications that we have today? So how is this matter being perfected? So because the pace that we have in the evolution of AI system can be followed by the evolution in the security systems.
Yes, it's such an important point. And, I think that every company that is in this space is investing very deeply in cybersecurity. At Medtronic, we have a really robust big group that is for cybersecurity experts to ensure that these models are developed in a secure way. But I think you are right, and it's a great question as we ensure for all of our connected devices, whether it's software or AI software, that we need to ensure the security of this. And it really gets to investing in our cyber security experts, ensuring that we are developing these secure models.
Perfeito. Bom, eu recebi aqui uma outra
Perfect.
pergunta.
I have received another question on our app, a question from the audience, which is, how can AI help in the risk assessment process and in the biological assessment process when submitting a pre-market license for a new medical device?
So just in the risk assessment process. So I think again, just AI can look across and analyze these, vast amounts of data. So if we have data on, for example, complaints or sort of adverse events and things like that, it can analyze these large amounts of data and be able to track and trend it in a way that we haven't done before. I was actually speaking to a colleague the other day about complaint handling AI, and she said, what would have taken weeks for one of my colleagues to do, AI can do in 2 seconds, because as long as you're feeding it the data, it can do these analysis. Again, it gets to making sure that we're feeding it the right data and the labeled data. But I think the same thing can be said with risk analysis, that it can start doing some of these tracking and trending over these adverse events and other information that we have to look at what we need to be paying attention to.
Alguma outra pergunta?
Any other questions? A microphone for you. Some time ago, I understood that in order to send a submission of an AI software, we would have to train the algorithm first, so it would learn everything. And then we would stop the training, and then we will conduct all the validations, even a clinical trial with the software, but it wouldn't be able to learn anymore. And then it could be submitted to the regulatory agency for approval. But that would be a software that would no longer learn as it was used. So is there the same perspective? Can we submit to an agency a software that is going to continuously learn with use? And if that is the case, from the regulatory perspective, how is this information, or how is the risk going to be interpreted, considering a software that is continuously learning? Cassie, would you like to start?
Yeah, it's such a good question. And something that actually, going back to predetermined change control plan, you can tell I'm passionate about that. Is something that is sort of thought about in that plan too, out of FDA. Because as the algorithms that are in medical device today, for the most part that I know about, are all locked, meaning that once it's on the market, in order to make the updates to that model, we sort of, as you explained, sort of shut down this, like any medical device, go and make the changes and then release it back in the market, versus adaptive algorithms, which they're learning from data out in the wild, out in the fields, and learning and changing in the field. So one of the proposals sort of put out there, but we, again, we're all learning. We don't know because there hasn't been an adaptive algorithm that has been put to a regulator, is this thought of putting guardrails around it. So you would go to the regulator and you would say, we will let our algorithm change within these certain guardrails. These are my universal sign guardrails, within these guardrails, and it can learn and adapt. But past that, it will be locked down and we will, make the changes. And so that's one of the things that has been sort of proposed in this predetermined change control plan concept as well. But even, speaking with US FDA, they said, we're going to have to learn as well the first time it's brought to us to see how we're going to best be able to figure that out. But that's been one of the things that we've sort of brainstormed on, of setting, some guardrails that you can sort of then allow it to adapt within.
Bom, só trazendo assim pequeno comentário a esse
Just as a quick comment on that matter, I'm not an expert on software or AI, but from our perspective, when a software submission reaches Anvisa, that assessment is done considering what we currently have, or what we have at the moment. And if the software continues to learn, in the end, you are creating changes to that original submission. So like Cassie mentioned, we need to establish a moment or a limit, a threshold, I don't know, to preserve that assessment at that specific point in time. And of course, later, manufacturers will develop new functionalities or changes in the software that will then be submitted again to regulators. So in my understanding, today, the way we work requires that, because of the limitation that we have on the assessment, on how the assessment is conducted today. But of course, one possible evolution is the PCCP that, as it was stated, this is something that still needs to be developed. And Anvisa is taking shy steps, not quite long strides, but we are trying to perfect our software regulation, but with a lot of care. We are being very cautious in this evolution, and I hope that at a certain point we are able to get to the use of the PCCP as well. Andrea, another question? Dr. Augusto, I think this, going back to this matter, so what is the possibility for the agency to have convergency in the use of a plan like PCCP? So the creation of a local regulation that would allow us to work with a predetermined change plan. Because Cassie mentioned the importance behind this model. We're not saying that this is the ideal model, but it's a model that has already been analyzed and developed. So is there any perspective on Anvisa's side, just like we are evolving to E-Star and other models, is there a plan to bring PCCP to Brazil? Well, of course that this is a goal to be reached. There is an intention to do that, of course, but it depends on a number of different factors. We depend on having a specific regulation for that as well. But anyhow, in any regulation that Anvisa produces, we try to follow what is being discussed inside IMDRF the most. And I think this has been perceived for quite some time now. And of course, if we had a work group that would allow us to develop this together with other authorities so we could bring this inside Anvisa, of course, the process would be simplified. But developing something internally is quite a difficult task, considering, of course, all the limitations that we face as Anvisa. But perhaps using models that are being applied by other regulatory authorities can really help facilitate the process. I would like to ask Cassie a question. Well, considering that AI, as was previously mentioned, has this goal of learning and evolving as it is used, and to a certain extent, it meets the interests of corporations, so they can keep up with the evolution of the market and generate resources for new investments. But considering citizens, individuals within that scenario, in your assessment, in your personal assessment, what are the ethical challenges that the system brings?
I think the biggest question is really was this trained and developed for someone like me? You know, if I am me as a patient going into my doctor's office and they want to use some model that has AI enabled in it, technology in it, was this something that was developed for my scenario? I have a chronic disease, and so it's very personal to me as well. You know, I take a lot of interest in my healthcare, and so being able to trust that your physician has the information to know whether or not it was developed for someone like you, and also that you can have access to that information as well. And so I think, from a big picture standpoint, it's really important for those that are in this space, for medical device companies, to make sure that they're really committing to meaningful transparency about these products. And I mentioned that Medtronic put out the AI Compass externally, and this was something that we committed to. But also, as through the development process, to make sure that you're thinking about, as you develop the technology for a certain intended population, and as is trained and tested on a certain population, that is provided in your labeling to make clear to, again, the physician or the patient. I just think that's sort of, in order to build trust, we need to make sure that our patients and our physicians are getting the information that they need for their healthcare.
Perfeito. Bom, eu acredito que...
Perfect. Well, I believe we are almost out of time, unfortunately. I just wanted to say thank you to Cassie Scherer for her presentation. Thank you for sharing your expertise with us at the seminar. This is a fascinating topic. And like I said, sometimes it is a little bit scary, but there is no way of avoiding it anymore. This is something that has taken over pretty much all areas of society. And when it comes to medical devices, this is certainly an evolution that can generate huge benefits. And it is a technology that will allow us to develop new drugs and new treatment options. It will allow us to identify even epidemiological trends. So it's truly an amazing thing to think about all the future that we have ahead of us with AI. So thank you very much, Cassie, for being here. and for actually traveling all the way down to Brazil, so you could share all the knowledge and expertise that you have with us. I hope that we have opportunities of working together again in the future with discussions related to the regulation of medical devices and AI and machine learning and all the new technologies that are here actually. So thank you very much.
Thank you so much.
Now we have a quick break for lunch, and we come back at 1:30. And the first panel will talk about validation and test for coded orthopedic devices. So at 1:30, I would like to ask you all to please be back so we can restart our agenda. There's also a housekeeping announcement by Dr. Sergio. Well, unfortunately, we couldn't change the agenda, but considering different flights that were canceled and viral issues, we had many issues to be faced. So this presentation on coatings will happen in the end of the afternoon, and the first talk after lunch will be on the clinical assessment of products. A presentation done by Rafael Braille with comments from Clarissa Rodriguez. So the panel that was supposed to be the last one will now be happening right after lunch. Thank you, Dr. Sergio, for the updates. Thank you, everybody, for staying with us. And let's remember that in the room next door, through a Johnson & Johnson and MedTech partnership, we have a workshop on the digital evolution on healthcare. Get to know more about the digital technology and have an experience in the metaverse. Very interesting workshop that required previous registration, but we do have some openings still. We come back at 1:30, and remember that all questions should be sent via WhatsApp. I count on you all to be back on time. Thank you so much.
Muito boa tarde. Solicito àqueles que ainda não entraram que, por gentileza,
Good afternoon. I would like to ask everyone to please take their seats. We are live at the 4th International Seminar on Medical Devices, promoted by the National Health Surveillance Agency Anvisa in partnership with the Brazilian Association for Medical Device Importers and Distributors. This event has been sponsored by Viman Sistemas. I would like to remind all of you that questions should be sent via WhatsApp on the QR code on the screen. We are going to start our first afternoon panel that will discuss clinical assessment in the development of valves and devices for cardiovascular surgery. In order to cover this topic, we will have Dr. Clarissa Rodriguez, Director from Green Consulting, to moderate the discussion.
no sofá. Para debater o tema,
And to discuss the topic, I also would like to invite Mr. Rafael Briley, an engineer and director of research and development at Briley Biomedica.
os microfones estão ali na lateral.
There are microphones on the table, and please, the floor is yours.
Boa tarde a todos.
Well, good afternoon, everyone.
É grande prazer estar aqui.
It is a pleasure to be here with you today. After lunch, I hope you all enjoyed your lunch. I sure did.
Gostaria de agradecer à comissão.
I would like to say thank you to the organizing committee for inviting me to speak today, but more than that, for their effort in preparing this seminar. The medical device community is a small one, but quite strong. So having the opportunity to discuss such relevant topics for our ecosystem is only going to make us even stronger. This is a huge opportunity for us to get to know each other and discuss all these important topics. So again, it's an honor and a joy to be here. Perhaps if we could show my presentation. Well, my name is Rafael Braille. I am Operations Director at Braille Biomedica. And within my department, I lead the product development team, so with a focus on R&D. And today I was invited to talk about the clinical assessment for medical devices. And since we work with cardiovascular devices, I will provide you with some examples on the development of stents and cardiovascular valves. But in practice, the clinical assessment process and its methodology can be applied to any type of medical device. So when we structured our clinical assessment process, the goal behind the process is to understand safety and performance information about the device. It's during this process that we get an understanding on the safety and clinical the performance characteristics. During the same stage, we map the possible adverse events that the device might have. And throughout the process, manufacturers should reach an equation analyzing the risk-benefits ratio and understanding if that is an acceptable ratio for that product to be marketed and for that product to remain in the market. So the clinical assessment is nothing more than the set of activities that are based on systematic and scientific methods. So it is a scientific documentation that is produced through an exhaustive analysis on the clinical data available for the device. And data could be available both in the pre-market stage of the device, and the main source of information at this moment comes from the clinical investigation on that specific device, or data can come on the post-market stage, when through databases, the scientific literature, we can find data that are available about the device, and of course, conduct a post-market surveillance process, so we can identify all relevant information on that specific device. And clinical data could be related to the device itself, the device that is being developed, or it could be clinical data that is relevant but come from similar devices or equivalent devices. Later in my presentation, I will give you more details on how we define equivalent devices. It's not enough for them to be similar products for you to, for example, have two stents. That is not enough. We have a number of technical, biological, and clinical criteria that are used, so we can consider one device to be equivalent to another one. Up until the moment I developed this, actually, up until now, I've talked a lot about clinical data, but what is clinical data? Basically, we're talking about information on the safety and or the performance of the device, information that is generated once the medical device starts being used. So every time a device is being used on a patient, we are generating clinical data that later will be systematized and defined. And when we talk about safety, we want to know if that device is able to deliver on what the manufacturer has determined without having an unacceptable adverse event for that specific device. And when we are talking about performance, we want to know if that device actually fulfills the function described by the manufacturers. So if we are talking about an aortic stenosis, once the device is used, it should be able to treat aortic stenosis in order to be considered a device with an acceptable performance. So, like I said before, clinical data can come through a clinical investigation on the device itself, meaning clinical assays or clinical studies, where we have a controlled environment where we clearly defined what are the patients that we are going to treat, what are the teams, the centers, and the clinical outcomes that we look for inside that specific trial. So it's a really well-defined and controlled environment. We can also identify data from clinical investigation or other trials that were published in the scientific literature from a medical device that is equivalent to the device that we are developing. And clinical data can also be generated through other clinical experience with the device itself or through an equivalent devices. And here we're talking mainly about post-market data. So when should we conduct a clinical assessment? Well, the process actually starts at the beginning of the product development. So when we come up with a concept of the device, the clinical assessment process starts together with the development of the concept. At that early stage, we don't have clinical data, and for that reason, we use a database from the preclinical assessment coming from biological mechanical trials or biocompatibility to understand what we will need to structure in our clinical assessment methodology. And then once we start to go ahead with the project of that specific device, we have a pre-market clinical assessment. And once the device reaches the market, while we have patients using the devices and while the device is available in the market, the assessment or the clinical assessment process needs to be ongoing. And we do that by using post-market surveillance tools and post-market clinical follow-up tools, which are the tools that we have available for that. And on that topic itself, we would require a separate presentation because it is quite a broad topic. Understanding all the structure that a company requires in order to have a post-market surveillance process in place. So this is the concept behind the clinical assessment process. And this concept is translated into a document, or which is the clinical evaluation report. And in order to build this documentation, there are some standards and regulations that describe the process of building this clinical evaluation. If we look at Europe, we have the European regulation on the matter, and we have MEDDEB Revision 4 that recently was published that describes the clinical evaluation process. And at Anvisa, we have Guide 31 from 2020 describing the steps of this process for conducting a clinical evaluation. These two references are quite similar in terms of context and content. They have a different format. They divide the process in different stages, but if you look at the general context, they are pretty much describing the same process to be followed. So when we start the clinical evaluation process, we search for ways of attesting to the safety and performance of the device. One alternative is using equivalent devices, and when we cannot find equivalent devices, we of course do a clinical investigation. And sometimes this might seem confusing. So how can I decide if a product is going to be compared to an equivalent device, or when should we conduct a clinical investigation? I'll talk a little bit about equivalence, but before going into details about that, we have a very positive experience in having conversation with the regulation authorities. Once we establish the rationale, if we're going to use equivalence or clinical investigation, it is strongly recommended to do consultations with Anvisa or doing pre-submissions with the FDA in order to choose or discuss the strategic pathway for that product and get feedback from the agency if that actually makes sense or not. So, like I said before, the process that uses an equivalent device follows a number of criteria that are quite technical. So, the device is compared with an equivalent device, considering some criteria. So, both devices, do they have a technical equivalence? Do both devices have similar designs or similar specifications and similar physical chemical properties? Do they have a similar composition? Are they going to be used under the same conditions, using the same methods and same surgical principles? If the answers to these questions are yes, we have a technical equivalence for devices. We also need to look at the biological equivalence. And when we look at that, it means that both devices should be in contact with the same tissues or bodily fluids. The period of contact should also be similar. And finally, the last criterion is the clinical equivalence. So both devices should be used to treat the same clinical condition. It's not good to have one device that is used to treat a vascular condition in a specific region and a different device that is used in a different region. So they could not be compared in this case. So it should be in the same area in the body and for the same group of patient by the same users. So, equivalence means that the medical device or the equivalent device should have a broad clinical use and it should contain high quality data in the scientific literature. If we have a device that is considered to be equivalent, is approved, and is available in the market, but it followed an approval process that doesn't provide us with enough clinical data, we cannot use it as a comparable device. And when we can find equivalent devices, this is a very interesting thing because it brings more agility to the process. It helps us improve the costs of a clinical trial. We know that developing a clinical trial is definitely the most expensive part of the development of a medical device. And later in my presentation, I will show you a comparative considering the Brazilian landscape in the costs of developing a device using equivalence and the cost of developing a device when we need to do a clinical investigation process. And besides that, it's also a good option for proving essential requirements because it allows us to understand adverse events and we can understand the risk benefit ratio and it allows you to learn a lot about your device before you start creating your own data. So again, a very interesting option. And equivalence data within our clinical evaluation report can be presented in different ways. One format that we find very interesting is this table that was developed by the Ministry of Health from Australia. So we place our device, the comparable device, and once again, we look at the clinical, technical, and biological characteristics of both devices. When we don't have access to any of these characteristics in the literature, we have to find ways, if it's the comparable device comes from your own company or a third-party company, you need to request access to the device in order to identify data that is not available in the literature. Once we have filled out the table, we add a third column to it, which describes the impact of the difference in the safety and performance of the device, comparing your device and the comparable device. And in this third column, we come up with a justification for that difference and why it does not interfere in the safety and or performance of the device being assessed. If that rationale can be concluded, that device can be considered to be comparable.
I have mentioned the two guides, the European and the ANVISA guide for the process of clinical evaluation. And so here are the main stages of the European guide. So the European one mentions the stage 0, where we identify the scope, the essential requirements that will need support from clinical data. So on this stage, we define the stage, the team, the assessment plan that will be conducted. In stage 1, we identify the clinical data available in the literature, clinical experience, or clinical investigation. So this systematic revision of data is conducted. And once we have data in hand, we start the process of assessment. So we assess the set of data regarding the quality of the methodology, the scientific validity, clinical relevance, and the weight of contribution. Stage 3 is regarding analysis. So when we evaluate data regarding the strength of the evidence, and on stage 4, we prepare the reports that show the compliance with the requirements. And there's a need of constant update. So this is updated with the processes post-market. Here we have the process in guide number 31 by Anvisa. So we have 3 stages, but if we analyze the content of these 3 stages, it's the same that we see in the European guide. So they are separated differently, but the content is similar. And so Anvisa has this fluxogram. So after the data analysis, we decide if the clinical evidence is enough to declare conformity with the essential requirements. If it's not enough, then we need to generate new clinical data or additional clinical data, or even conduct a new clinical study. If the documentation and the evidence meets the conformity, then we produce the clinical evaluation report. So this is the final stage of a whole process that is happening together with the development of the product. So what data do we need to collect? We collect technical vigilance alerts, data from clinical assays and other relevant clinical data, and then data from post-market surveillance. And the most significant ones are from post-market clinical follow-up. Data sources are basically the journals where we have scientific articles published. The main reference is PubMed, but these are all other portals where we use as reference, and regulatory authorities. So we can check FDA, MAUD, and other data sources to identify adverse events and the behavior of a certain device depending on the clinical use. We classify ideal data, those that are considered proper for the creation of a good CER. So it could be pivotal clinical assays data. So that's the best one when we have the device with a comparative. So the idea is not to show inferiority or superiority. The primary outcome needs to be of a clinical nature, and there needs to be enough follow-up time to identify both acute and late adverse events. It's important to have also randomized clinical trials. So that is very relevant. And we also have observational studies with prospective or retrospective cohorts with a relevant number of patients and a long period of follow-up, so that these data can also be used to build that report. So when we want to present the clinical evidence, we have 4 fundamental aspects. Data should have performance and safety clinical outcomes. The clinical evidence must be from the devices that are being declared the equivalents. And so they have to refer to that specific device. It's necessary to present clinical data for the indication of use for that medical device, and data must come from pivotal studies. So non-pivotal studies like cohorts may be used, but as a complement to your report. One of the questions that might come up for manufacturers and people involved in that process is, for which medical devices and when do we need to do that, this assessment? And so the answer is for every device we have to conduct this process of clinical assessment. This is not optional. This is part of the technical dossier, because from the clinical standpoint, this is the most important document about the clinical use of that device. So this is mandatory, and it's a definite tool for the regulatory authorities to make decisions about the safety and performance of the device. So here on Anvisa RDC 751, products class 1, 2, 3, and 4 have as a mandatory summary of the clinical evidence. And what does that mean? It means a technical dossier, and this summary of the clinical evidence is the report of clinical evidence. What is the frequency that this report should be updated? So the manufacturer defines and justifies the frequency of updates. Rule of thumb, products that have higher risk are updated with a higher frequency, with less risk than less frequent. But this is a definition defined by the manufacturer. And we see every 2 to 5 years, depending on the risk classification of the device, or whenever new clinical data come up in the post-market that would suggest an update on that report. These are some examples. So here is the development of a vascular stent. Once we started developing this device and collected data, we saw that this could be a device that could have this process conducted by equivalence. So this is a reality of the Brazilian industry. So this would be, the figures that we would see. It's a 5-year project, and the construction of the clinical evaluation report. The clinical evaluation report, it's the final stage of the clinical assessment, and that happens throughout the development of the project and happens continuously till the post-market. So we had 15 people involved in the project with other 5 employees for the report. This is a project that costs about 3 million reals, and the CER cost about 60,000 Brazilian reals. The report had about 100 pages, just for you to have an idea. This is the second example of a transcatheter valve. We understood with the manufacturer here in this case that we would have to conduct a clinical investigation with pilot and pivotal studies. And here I would like to highlight again that it was really important to be close to the regulatory agencies to define this process. So we presented the rationale to Anvisa to follow this path. This was a 10-year project with 5 years of clinical studies conducted. We have almost double the number of employees, especially for the construction of the report. So 18 employees for the project against 15 from the other one. From the development point of view, it cost more than 10 million Brazilian reals. And if we consider the clinical study part with the internal staff, this is a study that costs about 2 to 3 million reals without considering the contract research organization. And it's a more robust document that gets created. So over 200 pages in the clinical evaluation report. This process might seem very expensive and complex, but we are here for one goal, which is the patient. You know, everyone participating in this process deals with the reality of providing new solutions for patients. So life is always put in first place. So we always, you know, we have to consider this process because it's an important and systematic process to provide more safety for patients. Thank you very much. Rafael, thank you very much for your presentation. It was excellent. I would also like to thank Abride and Anvisa for the invitation. We have seen that this process of clinical evaluation have increasingly required scientific methods to be conducted. So you already have this process established. So how have you been dealing with this? What type of guidelines have you been using in your internal processes? Not only considering pivotal studies, but the whole process of selecting the studies, how to extract data, how to report a clinical outcome in a scientific way, and, you know, constructing a document in the quality that the agencies require. Right, Clarissa. So it's a systematic and scientific method. We use the systematic revision method. And yes, there are some guidelines that recommend the systematic review process. We use Prisma's guide, which is very clear about conducting this process, and there are other guides that can be used. So we use the one by Prisma and by Cochrane, which are very interesting and additional tools for the systematic review process. Perfect. I also enjoyed the way you compare, you know, clinical evaluation with clinical research. Even though the names are similar, the processes are completely different. And what we see in practice are companies getting to the end of this process of development without thinking about what the product would need for an equivalence approval. And so they get to, you know, a path of clinical investigation. So considering the equivalence, when do you start considering that for the clinical evaluation report? Well, since the beginning of the project, once we initiate the project, we already think about the process of clinical evaluation. And it's a sad surprise when we get to the final stage of the project and we find out that we would need to conduct the clinical investigation process. Because, you know, of course, you know, the time, the costs, the whole structure that you need to create to conduct that. And if that comes up as a surprise during the project, then that's, you know, that's really not interesting. So we already studied that strategy right at the beginning, from the get-go of the beginning of the project, so that we can, you know, see what paths we're going to take. And since we have a lot of importers here, people sometimes get surprised, you know, because they bring products that already have a clinical evaluation report that comes from abroad. And oftentimes when they get here, they have to conduct a clinical investigation. So it's important to have that type of conversation between manufacturers and importers, you know, to understand with the agency what are the processes that need to be followed to build that report here. That's important for us to prevent and avoid those unnecessary surprises during the project. Exactly. We see with a lot of frequency importers bringing products that were developed abroad, and then they get surprised because they will need to generate more data, and that requires time and money for the equivalence approval process, like you mentioned. We have some questions from the audience. Considering the updates of MEDEV that require more similarity, how does that reflect in submissions in Brazil and Europe that use the European seal? Well, regarding MDR, I think that's the debate for manufacturers of medical devices. We see that MDR continues to be the biggest regulatory challenge today, especially because it's new, you know. I think we will be able to have more clarity once companies, you know, get certified and the process gets more mature. But today, even with the notified bodies in Europe, it's difficult to define what your strategy will be for the technical dossier of your product, what will be the path to conduct such a clinical evaluation in Europe. And that's why most companies that have always had Europe as an entrance door for their devices, even before the US, have been revisiting their strategies and prioritized the US or other jurisdictions where there's more clarity about how the clinical evaluation process should be. So for us, it's been really a challenge. And I think, you know, as industry, we are kind of still waiting for new products to be certified in Europe so that we can understand how we can conduct this process over there. Right, our experience is exactly the same. We have seen a huge decrease in submissions in Europe. There's no impact in submissions for Anvisa, but for Europe, they're literally basically almost zero. It's a huge difference now. And we have another question. The impossibility of using a predicate that used the equivalence pathway to show safety and efficacy is something that is used by different health agencies. As far as I know, and I can only talk about our experience, a predicate, if it does not have clinical data, it cannot be used as an equivalent, regardless of the country that you wish to regulate the product. You cannot use equivalence only for the equivalence, you know, you have to have access to the clinical data in order to be able to use that device, regardless of the place. Exactly, same with us. So for example, you have the equivalent product, but you only have clinical data for some indications of use. So even if that product is registered, if you do not have access to the clinical data, you will have to narrow down the indications of use of your product to register it. So you will need the clinical data of the equivalent product for all the indications of use, and clinical data considering the parameters that Dr. Rafael already commented. Considering the revalidation of the record, is it mandatory to use the CER? Yes, you always have to have the updated documents of your clinical evaluation report, even when you're submitting for class 1 and class 2. Sometimes the manufacturer goes, oh, I do not have to send the documents. And that's not because you do not have to send the documents, that you do not have to have those documents. So sometimes you do not have to send the clinical evaluation report, but that report should be updated constantly and the manufacturer should follow the updates of the report throughout the life cycle of the product. Excellent. And we have one last question. Is it possible to use the chemical characterization ISO 1099318 depending on the application of the medical devices? So the use of it can facilitate the process to guarantee more safety to the patients. To reduce the number of clinical data, I don't think so. I'm not sure if Sandro, that works with regulatory affairs at the company, would like to add something to it. So this is safety for the manufacturer regarding the biocompatibility data of the product, and those need to be contained in the technical dossier of the product. But we do not use those data to reduce the number of clinical data that needs to be collected. I'm not sure if you would like to comment on that. No, I absolutely agree with you. It is mandatory. So you're not exempt of presenting clinical data within the parameters that we're talking about here. So those were the questions. Do we have any more questions from the audience? You have talked about the regulatory affairs. Would you like to have someone like Sandro add to what you just said? Please wait for the microphone to get to you. And I think we have another question from the audience here in the first line. Right, so that's the idea, what Rafael mentioned. Data from ISO 993/1819 will offer different data from biocompatibility, but clinical data need to be collected anyways. I think we have another question from a front row here. Just wait for the mic to get to you. Here on the first row, please.
My name is Deborah from Boston Scientific, and we have been hearing a lot about the new European regulation and that we don't need to access only the clinical data from the equivalent device, but more detailed project data. So it is pretty much impossible to claim equivalence. I think you're nodding as already answering my question, but the question is, what is your experience in reaching equivalence for these class 3, class 4 devices? Well, equivalence with itself, with our own devices, or with a company where we have a joint venture or a collaboration agreement. Because I think this is the goal, actually, to limit the use of third-party devices as equivalent. So you need to have a collaboration agreement between both companies. We have some examples of that. We have partnerships with some startup companies and some companies that are developing products that are similar or that could be understood as equivalent devices to our own. And many times when we are discussing these initial collaboration steps, we discuss the type of documentation that we will provide access to or require access to, depending on the scope of our partnership, so we can actually conduct or claim equivalency. But it seems to me that without the authorization from the manufacturer, we won't be able to claim equivalence anymore. Any other questions? That's it? All right. So, on behalf of the organizing committee of the seminar, I want to say thank you to Clarissa Rodrigues, Director of Green Consulting Brazil, and Mr. Rafael Braille, Director of R&D from Braille Biomedica. Thank you very much for your presentations. I would like to remind you all, like it happened in the morning, all the content being presented here today and tomorrow will be made available to all of you through an email that will be sent, so you can fill out the survey on the event and receive the material. I also would like to remind you that we have a very interesting workshop taking place right next door related to the metaverse. The workshop was developed by Johnson & Johnson MedTech and discusses the digital evolution in healthcare. So if you have an opportunity, it's very interesting workshop. Sterilization as a necessary requirement, how to deal with that. This is the topic we're going to cover now. To moderate this conversation, we have Dr. Nancy Mesas from Rio, from Assemble Regulatory Affairs Consulting in Brazil, whom I would like to invite to join the stage. And our speaker will be Emily Waller, the principal scientist from Boston Scientific, USA, whom I also would like to invite to join us on stage. Good afternoon, everyone. Well, good afternoon. First, I would like to thank the organizers for inviting me to moderate this conversation. I'm very happy to see a full house and to learn that so many people are watching us online, which really demonstrate how serious this topic is. And the discussions definitely have been great. Today, we have the privilege to welcome an expert on this topic, Emily Waller. She came from the US to gift us with her presentation and to share her vast experience and guide us through all the nuances of sterilization, which both scares us, but is undeniably important for everyone working with medical devices, because we know that most devices come in a sterile presentation. And speaking of reliance, we know that this is one topic that will have a major impact in the acceptance of tests, reports, and studies. So our goal here today is not to go into great depth on the topic, but to get this perspective from an expert. So the idea is to have an open discussion after the presentation, so you can answer any questions you might have. And please feel free to ask all the questions that might come up during the presentation. And again, if we're not able to answer them today, we will make sure you get the answers later on. So before I hand the floor to Emily, I would like to invite you all to enjoy her presentation. Emily, please.
Thank you, Nancy. Thank you for everyone for the opportunity to be here. So sterilization is an essential requirement. It's very similar to clinical, but I'll kind of take off from, from where the last speaker left off. So for sterilization methods, we have kind of a menu to choose from, depending which device and the materials and temperatures it can withstand. They're kind of broken down into a gas method or a radiation method. So for gas, we have steam, ethylene oxide gas, otherwise known as EO, and then a plasma or vaporized hydrogen peroxide. Liquid chemical sterilization usually uses a glutaraldehyde or formaldehyde, but the validation methods are similar to gas, just in a liquid form. With radiation, gamma has always kind of been the gold standard, but now with gamma capacity being very limited, we've moved on to E-beam and X-ray being viable options with a lot more capacity, which is critical to our manufacturing capabilities. So the standards that are used by all methods, just kind of as a blanket, would be the ISO 11737 kind of suite of standards. So the main one is bioburden. Bioburden has to be controlled for any method of sterilization.
That's kind of the basis of all our methods, to make sure our bioburden's in control. So that way we know the process we have validated will be effective on that bioburden. So the ISO 11737-1 standard tells you how to test your bioburden, how often, kind of gives guidance, and just general controls. Along with that is ISO 11737-2, and that is for sterility testing. So for a lot of methods, you actually will immerse an entire device and make sure there are no organisms left on that device that are alive and could grow. So those are the two main pieces that we would use for validations. Along with that, but separate from sterilization, is the ISO 11737-3, which is the endotoxin testing. And that falls along with biocompatibility, but responsibility for that usually falls within the sterilization group. So you can have a sterile device with endotoxin failures, or vice versa, a non-sterile device that has endotoxin. But endotoxin standards are technically separate from your sterilization validation. This is just kind of a cheat sheet, a reference sheet for the different ISO standards that would need to be referenced and followed for any regulatory submissions. So with ethylene oxide, you have the ISO 11135 that governs sterility, but you also have the 10993-7, which documents EO residuals and what you need to follow for that. All the other methods have just the one standard for sterility based on which method you use. The one exception is, so radiation, even though there's gamma, E-beam, and X-ray, they all use one ISO standard, the 11137. So that makes it a little bit easier that all radiation is done the same. So for overall, I'll start with the terminal sterilization as manufacturers would do. And all of them kind of follow a general pattern. So we all use a sublethal cycle for validation. All the gas methods and liquid chemical will use most often a half cycle method, where you'll take a defined time of exposure at a set concentration of your sterilant, be it EO, vaporized hydrogen peroxide, steam, or liquid chemical. And you'll validate that you have no growth of organisms after that half cycle. And then for your routine cycles after validation, you'll just double it. So it's a very simple, in theory, but straightforward, just calculated approach. There are other methods within the standards that you could use based on bioburden, different things like that in a pinch, but half cycle is the most common method. Each method has its own indicator organism based on whatever microorganism is the hardest to kill for that method. So for example, Bacillus atrophius is the most difficult to kill spore for ethylene oxide. So all testing for ethylene oxide sterility with biological indicators uses that organism. But for steam and vaporized hydrogen peroxide, it's actually Bacillus or Geobacillus, depending who you're talking to, Stereothermophilus. So that organism is harder for steam and vaporized hydrogen peroxide to kill. So all biological indicators will use that organism. And then for liquid chemical sterilization with glutaraldehyde, it's typically Bacillus subtilis, but Mycobacterium chelonae is also a concern from infections for patients. So you would use whichever one you have data to support being more resistant to your sterilization method. So for example, our heart valves, when we initially validated, we used Bacillus subtilis and Mycobacterium chelonae, and the Bacillus subtilis lived a lot longer in the glutaraldehyde. So that's the indicator organism we use for all of our validations, just to make sure we're using the most robust process possible. In contrast to the EO method or the gas method, radiation doesn't use a biological indicator. It will use a lower dose of radiation as defined in the ISO standard, depending which method you're using. And then you'll irradiate your product at that and then do a sterility test to make sure that your product is sterile at that sublethal dose as well. There are a couple different methods for radiation. Most common methods are the VD max methods that are based on the level of bioburden you have. So VD max 25 is the most common. If you have a really, really low bioburden, you can get away with VD max 15. And if neither of those methods work, then that's when people move on to what's called method 1 or method 2. But those used to be more standard, and they've been more commonly replaced with VD max. One other thing to note, excuse me, is that all validations need to be conducted in final packaging or packaging that is equivalent. So your same amount of Tyvek or same film or same density, that type of thing, just to make sure that your validation is representative of your final sale product. To lead into that, so the packaging type will be defined based on your sterilization modality. So for all the gas methods, EO, steam, and BHP, the gas penetrates the packaging and into the product on steam, which goes in through a breathable membrane. So those packages usually have Tyvek, and the amount of Tyvek is a critical feature. The more Tyvek, the better. The easier it is to get gas in and out of the product. Liquid chemical, main requirement is to have a rigid container that can contain your liquid. And then radiation has a lot more flexibility. You could use a film, you could use Tyvek if you want, but that's more expensive, or foil. Just it has to be able to have a good seal that complies with the ISO standards for packaging, and it has to be compatible with radiation. To break down a little further, overall, the process steps are similar for the gas methods. EO has a preconditioning phase. It has a preconditioning phase, and the goal of that is to increase the product temperature and humidity. Without a certain baseline of humidity for EO, you will not get the kill that you're expecting for your device. So humidity is a key component to that process. After your product has achieved the correct temperature and humidity, to get above that threshold, then it proceeds into the sterilization cycle, and it's exposed to the EO gas. It's more lethal. After the sterilization cycle is over, it will proceed on to an aeration phase where warm air is changed out in order to remove the ethylene oxide from the product and drop your residuals. Steam is of a similar mechanism. In the beginning, a vacuum is drawn to remove all the air from the product, and then steam injection pulses are happening in order to kill your device. So rather than using the EO gas, it's steam at a higher temperature. And then at the end, there will be a vacuum to withdraw the steam and dry your device, kind of like the EO. So process monitoring for routine sterilization is, again, bucketed based on which modality. So for gas methods, this didn't go. You'll use what's called a self-contained biological indicator, oftentimes just called an SCBI. And the bottom, I can do this. This bottom little white strip here is a spore strip. And so it's got 1 million spores of whatever organism is the most challenging to kill. The purple little vial is media, and then it's all contained in a vial. So the nice thing about this is it goes onto or into your load, depending where your validation said it made sense to put it. And then at the end of sterilization, you can pull it out and crush the little vial inside, and then that whole thing goes into the incubator all in itself. So there's no risk of human contamination or any risk to your indicator failing because of someone contaminating it. So it's a nice simple process. You can get 5 to 50 of them on a load pretty quickly and easily. They all go into an incubator on a little rack per load, so it makes it really easy to monitor your process. Radiation is a little different in the fact that you validate a range of dose. So there's a minimum and a maximum of dose that you'll have to validate. The minimum dose assures that the device is sterile. The maximum dose is the maximum that was tested to find a device that still functioned. Not that it won't function above that dose, but it's just the highest that we have tested to show that it functions. And then for your actual validation, we have to do what's called an inoculated product. Most common is a biological indicator strip. And it's the same as what's in the self-contained BI. It's just outside of the little vial. And we place it in the most challenging to sterilize location of the device. So wherever we think that if one microorganism was hiding, that it could live if sterilization had a hiccup, that's where we would put this. So in the case of the device that is shown here, that little crevice is capped. And so it's a dead space. The rest of that device is all within an open tube. And so it's really easy for the gas to get in and out. So the most challenging location of this device is in that little crevice. And then we place the cap on so that the gas has to get under the cap and into that little nook. So when we're done running our half cycle, someone goes in with a sterile forceps and pulls out that little strip and drops it into media. And if nothing grows, then we know our process is robust enough to achieve a sterile product. The risk with that, obviously, though, is whoever is handling that forceps could possibly contaminate the transfer. And so that therein lies the risk. If you can use a self-contained biological indicator, that human error is removed from the situation. So if you have a surface sterilized device, like a sterile drape or a bandage or something like that, the self-contained biological indicator is actually great for validation. If you have a pack of drapes, you can actually just bury that BI down in the drapes. And when you're done with your validation, you just open the pouch, pull out the vial, crack it, incubate it, and you're set to go. And then oops, sorry, I didn't realize that didn't go. And then the last one that is the trickiest is if you have a device where you can't place a biological indicator strip and it is not surface sterilized, like a valve, for example, or something really, really tricky with a hinge or something potentially. There's something called an inoculated product where you take a defined million bacteria in a certain volume of liquid and you place it into that location. Again, human error, clumps of bacteria, there are issues that can arise with that process, but if that's your only option, that's what you do. So for example, our liquid chemical sterilization for our valve, the most challenging location is under several layers of tissue right over that post on this valve. And so in order to effectively demonstrate sterility, we take 1 million Bacillus subtilis and we put it inside that little pocket. And then we drop the valve into its glutaraldehyde sterilization liquid and then run our half cycle. And at the end, we pull out the whole valve and put it into media that would allow that bacteria to grow if any of them survived. And if nothing grows, your half cycle passes and you move on to the next phase. Kind of a side note that I'll touch on, I think someone else is going into more detail, but as we get into more sustainability and reusable devices, there are improvements that we're working on making. So the ISO 17664 governs reusable devices. Last year, or 2021, I guess, a new version, -2, was added to call out non-critical devices. So those would be non-patient contacting or only contacting intact skin of patients. It was found that in certain facilities, consoles, screens, cables were potentially mechanisms for transmission of infections. And so the -2 standard was called out to make sure that any instructions for reprocessing those devices had validations behind them to make sure that we were making the safest product possible. So one thing to note, all devices that are reusable have to be cleaned before they're disinfected or sterilized. If the cleaning process isn't robust, you could actually hide or provide cover with that soil for microorganisms so the disinfectant or sterilant can't reach the microorganisms and kill them. So that's one key piece to highlight. And then after they're cleaned, then they proceed into disinfection or sterilization depending on the patient contact. So if it contacts intact mucous membranes, it only has to be disinfected, but they prefer you sterilize it. If it contacts blood, then it has to be sterilized. So those are kind of the breakdowns of those. Reusable devices can be sold sterile or non-sterile, depending on both the manufacturer and the user. Some users prefer to have a sterile device so they don't have to think about processing it before the first use. Other customers don't really care and are happy to sterilize it before they use it the first time. Steam and vaporized hydrogen peroxide are the most common methods for sterilization in hospitals. It used to be EO and steam, but now with the new environmental concern about EO, that's being phased out by most facilities and they're replacing it with vaporized hydrogen peroxide. Once vaporized hydrogen peroxide kind of breaks down, it goes into water and carbon dioxide. So it's much more environmentally friendly, fewer risks involved. So as we're all catching up with that, a lot of reusable devices have to be validated for vaporized hydrogen peroxide to meet the customer needs going forward, which usually requires more submissions and more for you guys. The other thing to highlight is that the processing instructions for reusable devices must be in the IFU. FDA for sure is watching those with a fine-tooth comb as the EU and many other regions as well. So that's kind of been a switch too, where in the past you could kind of have it available for customers if they ask, but now it's a requirement as part of the submissions. One other thing to touch base on is the biocompatibility impacts that sterilization can cause, mostly from EO residuals. In order to test the EO residuals, the devices have to be sterilized through the maximum number of sterilization cycles and the minimum amount of aeration to make sure they've seen the maximum amount of gas and the minimum amount of removal. So those are things to make sure you document in your report, what the rationale was for that, what those times were. The extraction time for the residuals is based upon the time of patient contact during a procedure. So if it's a 15-minute procedure, you really only have to test extraction for 15 minutes. Oftentimes we'll go for 30 minutes just for a buffer. In the past, the standalone was 12 hours or 24 hours just to be safe. But we found we were over extracting and pulling off more residuals than the patient was seeing. And so it was making our process a lot more difficult than it needed to be. So now we've gone to a risk-based approach and have made sure we've documented the maximum procedure time and then the amount of extraction time that we've done. And within that, the limits for ethylene oxide residuals are based on the patient contact. So the devices are broken into 3 categories: less than 24 hours, 24 hours to 30 days, and more than 30 days, which are usually your implants or your permanent devices. The other newer development is if devices are used in special populations other than adult males, there are reduced residual limits that must be calculated based on the population body mass. So an adult female would have 1 body mass. Children, infants, neonates, and preterm neonates all continue dropping down the intended patient weight. And based on that lowers the amount of residuals that you're allowed. So if you're intending on using your device in a neonate or a preterm neonate, you might have to do additional aeration to make sure your residuals are low enough to be safe for that population and not cause any irritation. Along with the biocompatibility impact of residuals is endotoxin. So any device labeled non-pyrogenic has to have endotoxin testing. There are a few different standards you can use for that. Most common is the ISO 11737-3. The interesting thing with endotoxin is it usually goes with sterilization because you could have a sterile device that you killed all the organisms on, but when those organisms were killed, their cell membrane lysed, and there's a toxin in there that can cause a fever in a patient, which is technically a biocompatibility reaction. So it's kind of a gray area. And so usually it goes to sterilization because sterilization causes those organisms to lyse, and therein lies your endotoxin issue. So each batch has to be tested. I mean, you validate your general process and your control of your bioburden for this reason, but then you have to continue to demonstrate that each batch of product meets these limits, which it's a serious concern, and that's why they're so particular with it, just like each batch of sterilization has to be tested for sterility. So the batch size is classified by the organization. And most commonly it's a week's manufacture. It depends. Some high volume products, they'll say it's a day, but usually it's a week. Devices that take weeks to build, sometimes it's a month's manufacture. So you have to document what your rationale is for a batch size and why. And then based on your batch size determines how many samples you need to test for endotoxin. So the minimum number of samples being 2 and the maximum number being 10. And then those all have to meet the defined limit for endotoxin. This looks really bad, but this is mostly a checklist for your sterilization report, which is usually 20 to 50 pages. So this actually isn't so bad for a checklist. But your protocol or your report, you can maybe document some in each, have to have your validation method, whether it's EO, steam, radiation, documented. The minimum dose that was tested, if it's radiation. So if it's whatever sublethal dose you ran, or if you'd used a gas method or liquid chemical, what your half cycle time was. Was it a 2-hour cycle? Was it a 4-hour cycle? Then you have to have your sterility test for radiation, showing that your devices all were sterile at your sublethal dose. Or if you're using a biological indicator for your gas method, you could use the bioburden resistance. So you'll look at the amount of bioburden that's on your device, and usually we try to cap it at 1,000 or so units or CFU on your device. And then you compare that to the million organisms that are on the biological indicator. And all million of those are the most challenging to kill. So it's a lot harder to kill a spore than it is to kill a staph that's on your skin. So if you've got 1,000 staph on your device, but your biological indicator has 1 million spores, obviously that's a more robust challenge to sterilization. And you don't need to do sterility testing, which is more prone to human contamination. In the past, you used to have to run both. You'd have to run sterility units along with your biological indicator units and show that you had more biological indicators grow than your bioburden. And after decades of showing that this was the case, it was kind of finally accepted that staph is always going to be easier to kill than your BI. So it was just kind of redundant. And that sterility testing for gas methods has kind of gone away. If you're using EO, you'll have to document your minimum aeration time, and then that minimum aeration time will apply to all of your routine sterilization. So you want to get that down to the minimum possible because supply chain really hates long aeration time. You also have to document your minimum sterilization time and parameters. So for gas methods, that would be temperature, pressure, and humidity. For liquid chemical sterilization, that would be your minimum sterilization time, your minimum temperature, and the concentration of your sterilant. And then for load configuration, you'll have to document your density, how many pallets you ran, locations of inoculated devices, all the different details that would impact your sterilization. So if you had a 4-pallet load, you'd want to know what the density was, what was in that load to make it a representative load, the density of that load, because that would be your maximum amount of product you're going to be able to sterilize on a routine basis, and where you put the inoculated devices or biological indicators to show that you got the most challenging spots within those pallets for EO gas or steam or whatever to reach. So you had your most robust process possible. If you're using radiation, you'll need to include your dose map that shows which doses were achieved all throughout. It's almost like a heat map throughout that pallet on the inside and all the corners, the highs and lows. So you should really show good control of your process and have a good understanding of that going forward. You also need to have the rationale for the device you tested, which inoculation method you used, and then the location of that inoculation, both in the device and within the load. The other thing that's heavily scrutinized would be your cycle records, so they can see which cycles you ran and the parameters that were achieved. And then the other piece of this that is often forgotten is sterilization validations will validate the minimum process to achieve a sterile product. So like everything, there's a minimum and a maximum. So we validate if you run a 2-hour cycle with 12 hours of aeration, you have a sterile product and it meets EO residual limits. However, you can exceed those times too and go up to longer cycles and longer aeration times. And at that point, its quality will show that the device still functions at that level. So we're on the minimum side to show that it's safe if you go to the bottom of the process. And that's mostly what we worry about in the sterilization report. Some common validation issues that can arise and kind of throw a wrench into things that we've seen are a positive sample, which would be growth of a biological indicator or a sterility sample. In this case, we would identify the growth to make sure it's the indicator organism and not lab contamination. It's surprising how often you get growth, everyone panics, they identify the organism and find out it was a Staph aureus or something that probably came from a skin cell that flaked off of someone's face as they were transferring a sample or floating in the air. So oftentimes what starts as panic is really no big deal. If you identify that organism and find out it's one of the bacillus that you intentionally added into there, then you need to check your cycle parameters. Sometimes you just barely missed your minimum time or temperature, or something was off with the concentration, and then you can rerun the cycle because it didn't meet your requirements for the minimum cycle. If your minimum cycle parameters and concentrations were met, then that's a true sterilization failure, and you need to go back and optimize your sterilization process. You might need to increase your time or your temperature or your sterilant concentration, the dose or your method. According to the standards, you kind of go back to the standard and it shows you what to do. So one thing that came about a couple of years ago with all the environmental regulations is the reduction in EO emissions. And so most companies went back and revalidated all their cycles with a lower ethylene oxide concentration. It helped us meet residual concentrations, but it also resulted in much lower ethylene oxide off-gassing, which was much better from a regulatory environmental perspective as well. So we're a little bit in need of the sterilization to remain able to use ethylene oxide. And so we did everything we could as manufacturers to make that process as safe for everyone as we could. And we've made a lot of progress with that. Sometimes we have to increase the time a little bit when we drop the concentration, but generally there were things we could do to mitigate the risk. So I think that's one of the newer outcomes going on. The other thing to point out is piggybacking on the last presenter, Product adoption is another option we have based on your modality. So you want to approach product adoption similar to how clinical does. You'd base it based on use, design, packaging, materials, lumens, challenges, those types of things. And a product adoption many times can be done, so you don't have to do testing on every new device if it's similar to one you already have. We set up product families, and then we base the families on the worst case, longest lumen, least amount of Tyvek, smallest lumen, those types of things. And then we have a rationale compared to our biological indicators to kind of assess risk for the process. So there are definitely ways to avoid doing months of testing for each new device, but sometimes you also need to do additional testing as you have more difficult and elaborate devices too. So it's not always possible to adopt, but oftentimes it is, which saves a lot of resources and a lot of time and risk. So with that, I think that's all I had.
Muito obrigada, Emily, por sua apresentação.
Thank you so much, Emily, for your presentation. It was perfect and really showed us what we wanted to discuss today. Wow. Yes, it's really technical, right? It's a difficult subject, I know. We even kind of get scary. I come from the technical area. I have worked a lot of time with radiation and EO sterilization, and I completely understand this issue as one of the main requirements for safety of a medical device. When we talk about sterilization, even if we subject the product under sterilization, we cannot guarantee that it's going to be sterile. So going through a sterilization process does not mean that the product is sterile. That's what Emily talked about, that guarantees that the product is indeed sterile, and it's not easy to talk about that. When we talk about Emily, we are talking about a person, an expert of the technical area and not the regulatory field. And so my greatest curiosity and my first question to you, Emily, is how easy or how difficult is it to deal with other areas that are not technical, because your subject field is something that gets the attention of the regulatory area, because when we say that we put on a label that stating that the product is sterile, if that's not true, then my product is not going to be safe and efficient. So my first question for you is, how do you deal with the regulatory department of the company and from the health agencies? Because a lot of times we get documents from manufacturers because we are not always the manufacturers. And sometimes the sterility reports or the validation reports, these reports are really difficult to read, to understand, and they are very difficult to meet the requirements that come with them. So how is it for you to, deal with all of that and to make everything that you work with be clear for the regulatory agencies?
Yes. So I've been very lucky in that most of the regulatory coworkers that I have been exposed to sterilization. So they understand a lot more than most people do. So they know what questions to ask. But we have been in situations with devices that we have not built, that we've received the sterilization report that we've had to say this isn't okay. And we've had to go back and retest. And that's not always a popular opinion. But if we don't have the data to support that process was robust enough to meet the regulations and the ISO standards, we don't really have a choice. It's pretty black and white sometimes. And so that's been
the go-to is we go back to the standards and say, this is either okay because it meets this, or it's not okay because it missed. And here's what we missed, and here's how we're going to close those gaps and move on. So as long as we've mapped it out and had a scientific rationale behind it, it's usually been okay. They don't always like the timelines that go with it or the cost that goes with it, but the risk is such a great risk that it's pretty easy to make the case for doing the right thing.
Perfect. Thank you for your answer. Obrigada por sua resposta.
Thank you for your answers. I forgot about the English word. Well, considering everything that you just said, you talked about the standards. So if I'm in compliance with the standards, if I'm meeting all the basic scientific requirements, I have answers. And I can answer my questions and talk about the requirements, even though I might not be the manufacturer. Because even if I'm not the manufacturer, I need to respond for that product to which I have the registration for. So you show to us that there are different standards, different regulations to be followed in the sterilization. Do they change quite frequently or not? The different ISOs, the different standards that support the sterilization process.
They're updated usually every 5 to 10 years. So they're always evolving, but sometimes they're just reaffirmed. Sometimes they say nothing new has happened that we realize, it's okay as it is, but they'll put a new date on it to show it's more recent. So there's not, over my career, there have only been a few major changes where they've added like the VD Max for radiation. That's been newer in the last 15 years or so. They don't change significantly too much, but guidances and little nuances or little ways you can do things will change. And generally anyone in my role within a group will have people reporting back. So we all change our organization SOPs as everything is updated. And then every time one of those is updated, we have to do a risk assessment against our SOPs, saying, are our processes still up to date? And if the regulations changed enough that our processes don't meet those, then we have to go back and look at everything else and possibly revalidate. And I can't remember a time of that being a case, but there may have been one that I just wasn't involved in. So they're always keeping state of the art, but it's usually providing additional guidance and making it easier to apply them and making it easier for others to understand who don't do this work every day. Those are usually the changes that are coming up, or just little changes, like when the ethylene oxide became a concern. There was some additional guidance as to when you drop the gas concentration, how to do that. There's discussions and trainings on that. So it's more just to help folks like me do my job better when those come out, but nothing significant. The new changes, I guess, are going to be like the vaporized hydrogen peroxide. That's newer in the last few years. And that is looking like it's going to start replacing ethylene oxide as much as we can for non-lumen devices. And so those will be the regulations to watch as those evolve, I think, as we learn more about that technology.
Perfect. Então, nós temos aí processo que...
So we have a process that doesn't go through many changes, right? It is a process that is systematically reviewed every 5 years, perhaps, the standards that support the topic, and changes, when they do happen, they are not so significant. I just wanted to add a comment and perhaps a second question. I'm part of the Biological Evaluation Study Committee, and we are working on the review of Part 7. We work twice a week in building this new text, and we have submitted it to analysis. We have received around 390 comments on our proposed text, just so you get an idea on how everyone is keeping an eye on this topic because of its relevance. Am I right? So we have received a number of questions from the audience. But before I read them to you, this is one thing that we actually discussed before together with Deborah about the sterilization of used products. And a number of questions about that, like we anticipated. So perhaps I can read the questions, and many topics related to that, to the reuse of devices.
Can I validate reusable devices processing based on literature?
Can you repeat the last part?
Eu posso revalidar
May I revalidate
produtos usados
reused products only based on a literature review?
o processo de
So the
esterilização
sterilization process for used products, can this process be validated
no reuso somente com
only by using literature data?
So are you talking about customers who want to use a single-use device and revalidate it?
nós podemos considerar que
Yes, we can. A single use could not be reprocessed. So perhaps a product that is going to be reused.
Okay, so like a cable or something like that. So no, we typically will use the ISO 17664, and that will tell us what classification you have to classify your device. Is it patient contacting, non-patient contacting? What part of the patient does it contact? And then that'll tell you what you need to validate. So you always need to validate cleaning and then disinfection and sterilization. So again, it's a very prescriptive process that the literature is there to tell you what your options are, but it won't tell you if your device is safe or not. Only a validation can do that. And then you have to make sure you use the correct soils and organisms. And it's a very cookbook type approach of what you need to do, which is really nice for rule followers, as to how to validate that product and make sure it's safe. And the other thing people want to do is make sure that whatever they validate is what their customers would use. If they don't have a washer disinfector, but they can only have a sink, you need to make sure you have the correct methods for cleaning and disinfecting the devices too.
Em outras palavras, para eu poder garantir que eu estou
In other
esterilizando
words, to make sure I'm sterilizing a product, I need to have as much knowledge about the product, its bioburden, the cleaning product, what are the microorganisms that are present there in order to ensure the validation process. So I need to know about microbial burden, resistance. I need to validate the process. And I won't be able to find information in the literature about my product's condition specifically. I need to know what are these conditions as soon as I'm submitting the process, right? Still on the same topic, there is a question here. Is it mandatory to do a validation for non-sterile devices that are sterilized by the user? So I distribute a non-sterile product and the sterilization process will be conducted by the user.
Yes, the same requirements for ISO 17665 or whichever method you're using or having your customer use would need to be validated. So very similar to how the manufacturer validates sterilization for industrial methods, we would follow that with the equipment the customer would use. So if they're using a steam sterilizer, we would use the same biological indicator strip, put it in the worst case location of the device, run it through half cycles, show it can be sterilized, and then we give those instructions to the customer as though we give to our teams as well to use. And so they would just follow those instructions, still use the biological indicator in their processing facility with each load like we would. And then they're all kind of taking over the manufacturer sterilization responsibility, but we're giving them all the data and the instructions to do so safely.
Perfeito. Então, temos que seguir as instruções do
So
fabricante.
yes, we should follow manufacturing instructions when they deliver us a non-sterile product. They give us these instructions on the sterilization process, and certainly that those instructions have been validated before.
Desculpa, gente, aqui toda hora volta. Outra pergunta.
Another question.
Pensando no aumento de
Thinking about the increase of the use of robotic surgeries where we have forceps that can be reused like 20 times. And thinking about such complex devices, how can we, nursing professionals, validate the cleaning inside institutions? Since today, the only test that is available is ATP used by endoscopy. Shouldn't these devices be able to be disassembled? So we need that support and do another public consultation, just some food for thought. We cannot assess biofilm inside the sterilization centers.
Eu falei, que assunto de gente grande, né? Não era bem o nosso foco estarmos falando de reuso, porque realmente é um tema muito polêmico. Então, eu até nem vou judiar da Emily, esperando que ela responda essa pergunta, né? Ou você quer... Não está ouvindo?
No,
Eh,
I got the first part.
okay, they are talking about
Estão
to
falando sobre
submit some products that can
alguns
be
produtos que podem ser
reused
reutilizados 4, 5
and
vezes.
they are have some difficulty to do this because they don't have information about the product, they don't have confirmation that it's possible to remove the biofilms.
Okay, so the manufacturer should have validated, and that's the beauty of the standards. They just released the ST98 in the US, AMI standards, which usually go into the ISO standards as to how you need to soil your devices to mimic how they're used. And then you have to clean them according to your instructions, and then actually put them through a process to extract anything that you can't see. And it has to meet an acceptance. So if those 4 steps are validated, and they're validated for 20 uses, for example, if you use them 20 uses or less, that manufacturer has assessed that they will be safe if you follow their instructions. So after cleaning, then it goes through disinfection or sterilization to make sure that they're safe for reuse. So that's why those instructions are there and why they're so precise in how we need to validate. We need to validate for 20 or 40 or however many uses. We have to validate at the end of that, at the minimum time, temperature, detergent concentration. We have to do the worst case. So you still have to look and make sure it's clean. But if it is clean as it looks and the manufacturer instructions are followed, it should be safe.
Yeah, it's not easy, okay? No. It's necessary for all sterilization methods, it's needed to define the SAO.
Yes. So the standard SAL is 10 to the minus 6. I don't know of many cases where you could do any less than that. Usually our SAL is more than that if you actually calculate it out. But if you follow all the sterilization standards, your minimum would be 10 to the minus 6, which is what the expectation is.
The residual toxicity is evaluated only for EO residuals. Cobalt, irradiation, steam requires residual.
Nope. Nothing's for steam or radiation, which is a lot easier.
We don't have residuals in irradiation sterilization.
Nope.
EO. Just EO has a part in the ISO 10993. Okay.
And that
Just
is the
EO.
one standard that I forgot when you were asking changes. That one, it always used to be based on the weight of an adult male. And now they've realized that not everyone weighs what an adult male weighs. And so they've changed the acceptable limits for ethylene oxide based on your population. So that has been one of the bigger changes I've seen lately.
And we are trying to specify the limits for don't just for adults, but neonates.
Mhm.
Yeah, it's difficult. Nancy, talk about mais próximo do microfone. Desculpem, veio a mensagem aqui para mim.
I got a
agora.
message. I got a message that I should speak closer to the mic.
Now, may I speak in Portuguese? Okay. Tendo visto
So
que está
considering that we had in the state of Paraná, we have a higher use of VPH than EO. I worry about the use of VHP, especially for cannulated products. What do you think about that? Using VHP for sterilizing cannulated products
We
or
are just
cannulas.
starting to get into VHP, so I am not as versed in VHP as in other modalities, but I know there can be some challenges. As long as the validations were done correctly with the biological indicator placed in the correct location and the standards were followed, it should be safe. But I think that's one area that we'll need to continue to watch. The vacuums aren't quite as deep, and so we're still learning with VHP. But if the validation data supports it, it should be okay. That's why we have the biological indicators in the process.
Ok, thank you. A compatibilidade
The
do VPH
compatibility of VHP with medical devices, different raw materials and packagings also hasn't been giving good results, which is another area to be discussed, something to be considered and study, right?
I think we're still learning, which material compatibility. There are AMI and ISO standards on that. And that's something we as device manufacturers are continuing to watch and test to make sure that we only use VHP with the devices that are compatible and have good results.
Okay, thank you. Emily, você teve alguma
Emily, do
experiência?
you have any experience? We don't have any more questions in the app, but if you want to ask questions, just raise your hand that a microphone will be taken to you. But regarding the presentations we had in the morning about eStar, have you ever had any experience on using eStar with sterilization data?
We have not, I don't believe that Boston Scientific has been a part of that, but that would be for my regulatory counterparts to answer. But we have templates, so almost like our own eStar process, where we have templates for our regulatory agencies that we tend to follow for all new products. So the same components and the same format is followed consistently, and all of our reports feed into those to streamline the process as much as we can. We would welcome a format like eStar to make things easier, but we're not there yet, I don't think.
Ok. Que é uma curiosidade, né? Porque
I was just curious because feeding this system with sterilization data from the process, that would be interesting, I think. Any other questions? Oh, a question there. Good afternoon. There are some sterilization methods that allows us to say, especially in the US and Europe, that are stopped being used, like ETO for environmental issues. You mentioned gamma radiation as well. Do you see any sterilization method that was not previously used, or perhaps a new technology that is gaining momentum that could be a method that perhaps has greater potential for replacing the ones that are stopped being used?
So ethylene oxide, there have been several sterilizers who have been closed down if they don't meet environmental limits. But overall, ethylene oxide for manufacturers has still been acceptable. It's just been very, very carefully watched. It's more in hospitals that they're not using it anymore, as the manufacturers of those EO sterilizers have moved to the vaporized hydrogen peroxide. Obviously, vaporized hydrogen peroxide doesn't work for everything that was EO'd, but it does work for a lot of cases. So that would be one to watch going forward. Gamma is still used. It's just because of the limited supply of cobalt-60 and facilities that can gamma irradiate and the amount of time that it takes to get a facility up and qualified. The gamma capacity, at least in the United States, is pretty much all accounted for. There's no way to get a product into gamma because there's no spot. So it's not that it's being phased out. It's just we can't create any more capacity. And so in its place, people have started looking towards E-beam and X-ray to sterilize devices that would have gone through gamma. And so with that, then you have to look at how your device and your materials perform in those modalities. So we had a device recently that would have gone through gamma, but we didn't have the capacity. So then we looked at X-ray and E-beam. And E-beam had a lot more discoloration of the product, and X-ray, very limited discoloration. And so we were able to modify the material to limit that discoloration and move forward with X-ray. X-ray is less efficient than E-beam, and so it's more costly. And so typically, if you can't go into gamma because there's no capacity, people will look to E-beam first and then move on to X-ray. So there's a chance that maybe there will be another radiation modality in the future that we haven't discovered yet that would be a replacement, but I'm guessing that's a number of years away with all the regulations and things that would follow. So you'll see more steam potentially coming up if the devices can withstand it. That would be another good replacement for EO, but it's also higher temperature, and that comes with some limitations as well. So it's nice when groups come to us in the beginning of the design process for a device and say, here's what I want to sterilize. Here's what it can withstand. Here's the conditions of it. Do we have anything else similar to it so we know where to start? Can we adopt it? Because sometimes it needs a design change or a material change or things like that. We've had to add stabilizers to plastics
to be able to not discolor in radiation. So there are tricks we're learning as we go to optimize our sterilization capabilities.
Realmente não é um assunto fácil, né? Então eu
It
agradeço.
is really not an easy subject, right? So I would like to thank everyone here for their presence. We have tried to present such a complex subject and talk about it as a essential safety requirement. It's mandatory within our process of validation of sterile medical devices. So I would like to thank Emily for presenting the different types of sterilization methods used, the main requirements that we have to consider, and I would like to thank her for sharing the technical standards. These are essential for any matter that we deal with in the regulatory arena, especially when we're talking about sterilization. We've had here someone that has the experience from the manufacturer point of view. So with that, we're able to understand this such a complex subject and the care of the manufacturer. And we cannot think about conducting sterilization processes without following all the concepts and precepts that the manufacturer follows so that we can offer safe and efficient products. We have talked about the sterilization and reuse of products. We talked about the responsibilities and the reports. And if the reports are not conclusive, then we have different requirements ahead of us. So we have to understand how to read these reports. We have to see satisfactory outcomes. And when we do not have them, we have to know how to explain them. We have to know how to investigate when we find any adverse events. And so everyone, it's such a complex subject. I would like to invite you to read more, study more. We have to deal with that by continuous studies. We are afraid of not using EO in the future because there are some products that are only compatible with that type of sterilization, but we're talking about a toxic substance that it's teratogenic, cytotoxic. And so this is something that has been very much questioned. And so we have a lot of things being talked about, and we even have the possibility of not using EO as a possibility. So just like Emily mentioned, we would have to discuss new materials even, because there are some materials that are only compatible with EO sterilization. So this subject not only concerns sterilization, but also material development and product development. So I would like everyone, I hope everyone enjoyed this presentation. I would like Emily, thank you so much for coming all the way from the US to share your expertise and your knowledge with us. And I wish you all a great afternoon and a good end of the day with more exchange of information and experience. Thank you so much, Emily, again. So we would like to thank Emily Waller and Nancy Mrs. Doria for their presentation. We now have a simulation of an orthopedic surgery happening in the metaverse room here with MedTech in the room next door. And this will be open for anyone who is interested in watching it. So now we will have a short 15-minute break, and we will be back to discuss about the challenges of efficacy and efficiency of implants. See you in a bit.
Olá, peço a todos que, por gentileza, procurem seus lugares. Vamos dar início.
Hello, everyone. So we ask you to please be seated because we are going to start our following presentation.
Boa tarde novamente.
So good afternoon, everyone. So challenges in proving the safety and efficacy of implants is our next subject. The conversation will be conducted by Priscilla Consigliero of the General Management of Technology, Medical Device Technology with Anvisa. Welcome, Priscilla. Our panelist to talk about this is Mr. Helmut Link, Managing Director and President at Waldemar Link from Germany. Welcome to the stage.
Ladies and gentlemen, presidents from Anvisa and from ANVU, I'm very happy to be here. Thank you very much for inviting me. And yeah, I like to give you my presentation. Maybe it gives you some ideas also for the Brazilian regulatory.
Boa tarde. É prazer agradecer
Good afternoon. We would like to thank Dr. Madeira, Dr. Sergio with Abride. I'm going to moderate this important presentation that will be conducted by Professor Helmut Link. I would like to summarize quickly because Dr. Helmut Link's summary is quite long. Dr. Helmut Link is the owner and director of Waldemar Link, and he is also a CEO of the Waldemar Link Group. He has a personal history in the orthopedics segment, and he participated in the development of the first prosthetics in a German hospital. In 1972, he launched his own plant, and from there on, the Link Group developed and expanded throughout the world. They have several branches and over 1,000 employees working with him. He continued to develop prosthetics, and today he is involved in the development of customized prosthetics, orthopedic prosthetics. So he's going to talk about the advances and challenges regarding safety and efficacy of orthopedic prosthetics, and that's going to contribute a lot to our learnings.
The rare species which still owns his company. And as such, I'm involved in the whole business, in the whole setup, and also, of course, in the regulatory. Before I start this presentation, I like to give you, I like to give you some of my experience when I first came across total joint prosthesis. I'm one of the older generations, and I started the business in the 60s. I learned my business as a surgical instrument maker. And my father then sent me to the US to see how business is done in America. And when I came back, I came across the first total joints. Here you see me in 1964. That's me, that's my father. And so I entered his office, and he had a big oak desk. He put a drawer, and he took a box from Brazil cigars and opened it up and said to me, "This is what we make for Professor Buchholz." And what I saw were hollow components like cups. And I thought, ashtrays. So I said to my father, "Okay, we make ashtrays?" "No," he said, "we make acetabular caps from polyethylene." Next day, I went to the hospital, the St. George Hospital in Germany, and I saw how they implanted in 1964 the total joints. Buchholz used the Moore prosthesis for the femur and used produce the acetabular cap. You see it here on the table. Now, the acetabular caps were not gamma sterilized. At that time, it was no gamma sterilization. So they were put in a bowl with a disinfectant for 24 hours. And then they were taken out and rinsed with distilled water. The cement, the bone cement, came from a bottle with powder, the polymer, and a bottle with the liquid, the monomer. So they took the powder, they took the monomer, and they mixed it. And that was autosterile because it generated heat. So they told me the heat sterilizes it. So that was how the original joint prostheses were implanted. The first joint prosthesis Buchholz implanted in Germany was in November 1963. Okay, now let's go to the topic. First, a disclaimer. The content of this presentation is solely based on our experience at Waldemar Link, which we have made in recent years in order to submit and approve products of medical devices with ANVISA and other regulatory authorities. They do not reflect any publication or study results, and we do not claim a complete overview. We not claim a complete overview of all possible submission experiences with ANVISA. Who are we? We are an owner-managed company with a long history, 1963 in implants, in the field of joint implants. Worldwide operating with implant systems and approvals in numerous countries. Many years of experience in the field of medical device registrations. Subsidiary in Link Brazil on site as an importer and contract to ANVISA. What kind of medical devices do we produce? Mainly orthopedic bone and joint replacements and osteosynthesis implants. Portfolio includes device for hip, knee, shoulder, and respective instruments. Class 1 devices, reusable surgical instruments, as well as 2B and Class 3 devices. What we use, metals polymeric materials, several coatings, and we do also EBM machining. How important is the registration of our medical devices in Brazil? To make it short, very important. CE market is our focus, of course. We are a European company, so mainly our main markets are in Europe. Brazil is an attractive and desirable market for orthopedic implants. It's a big market. It has a high priority for us. Within the regulatory affairs team, which we have 13 employees, 3 engineers take care of the registration in Brazil. Regular meetings with the Brazilian subsidiary to plan and coordinate regular activities and submission. As a comparison, for the US, we have 4 engineers, and for China, we have 3 engineers. A Brazilian registration can facilitate further approvals. That's another point why it's so important. Further approvals, mainly in South American countries. A number of manufacturers shy away from the effort of obtaining regulatory approval due to some difficulties of estimating success. Manufacturers have to comply with many regulatory requirements worldwide. However, there are still many country-specific requirements. ANVISA, as one of the largest authorities, could contribute to standardization in the approval process. I'm very happy that I recognize that ANVISA is working in the MDSAP program, which is a standardization of worldwide regulatory affairs. And this would help not only the regulatory institutions, but also the manufacturers to make things easier to standardize procedures, easier to understand between each other what's going on, and easier also to accept other approvals. GMP certification by ANVISA is a good accomplishment and positive sign internationally. In terms of complexity and individuality, approval in Brazil is more challenging than EU and USA. There is a little lack of publicly available information. The approval of innovation is based too strong on applicable standards. Nothing against standards, but there are sometimes new technologies where standards are just not there. And then we need to discuss engineer of regulatory with engineer of manufacturer. Is this product safe and efficient for the patients, for the population? Manufacturers have only limited orientation of how to handle such situations. Now, our in-house philosophy and future view. Challenges of the safety and efficiency of orthopedic implants and their durable function in patients are defined by the surgeon, the surgeon's competency, the patient's compliance, and the implant quality. And here again, the implant quality is basically a matter of design, material finish, and most importantly, the manufacturer's ethical attitude on his responsibility for his product. And that's why I'm also proud to be the boss and owner of the Link Company. Let's look into the modern joint replacement, present challenges of implant quality, new and future challenges, price quality controversies, and manufacturers' view on regulatory scopes. casting, forgings, and bar stock are ordered or are got into the company by these 5 steps. First, we have to define our requirements. Then we look at the supplier of material, for instance, and we check whether the supplier's certificate meet our requirements. Then we purchase, and following delivery, we do our own evaluation, chemical analysis. And if all specifications are met, material is released into production. Of course, measurement values are checked. Many other things are checked. And this shows you, for instance, I have my own casting company in Berlin, which casts cobalt chrome, titanium, stainless steel. And this is from the casting company for the first optical control. And you see, we have rules where to look, especially which parts of implants are under special stresses, special loads, in danger of breakage or whatever. And these are mentioned in rules which we give out to our workmen. And when they control, they know exactly, we have to look here at the stem in this area. This is very important. Or we have to look in the inside of the femoral component in this area, especially. And what we also do, this is a test or an X-ray control. Every part which we cast, for instance, is X-rayed in a batch, but the batch is numbered. So if something happens with one part, we can go back to our X-ray file and say, this part. Now let's look, is there anything to see? Did we miss anything? Then we also test several times a year our machinery. For instance, these casted parts are hipped, hot isostatic pressing. And the pressing unit is a very special setup, a special machinery, and we have to check, does it work properly? In order to do that, we put artificial defects here, for instance, in cobalt chrome distal femur components, or here, artificial gas defects in titanium components. And then we hip, and afterwards it has to look like this. The voids have to be compressed. So this is done, let's say, 2-3 times a year to find out if the procedures are working properly. Another test, very well known, is the crack detection with fluorescent dye test. So things which cannot be seen optically very well, especially in metal parts. These parts are covered in a fluorescent dye, and under ultraviolet light, one can find out or can see defects which are not found otherwise. Polyethylene. All the polyethylene worldwide, the medical polyethylene, comes from one company, Celanese. And then it's distributed to suppliers like Mitsubishi and others. And we get the polyethylene from these suppliers. What do we check? We check the measurements, we check the cleanliness. And here you see how they do it, how Celanese does the diversion of the different kinds of polyethylene. Here's the polymerization of the poly, and one track goes into industrial grade general industry poly. The other track with special purification. And as a special medical implant grade powder goes into the direction of the health industry. So one cannot just buy poly. It could be or would be probably just the industrial grade. That's no good. We want the purification of the poly material. And here a comparison of the essential requirements for poly medical grade. These are all requirements for medical grade poly. It starts with the quality management system, the change management, traceability, purity. For instance, calcium stearate, which is used in industrial polyethylene, is not allowed in the medical grade. The biocompatibility has to be all right. So it's a different table of rules for medical grade poly. Now, when we produce or develop something new, how do we look at the material? An implant must be chosen according to its physical and implant material, according to its physical and biochemical characteristic, depending on its best performance for a specific requirement. This is sometimes from some manufacturing company put aside in spite of the fashion. You may be, some of you remember, there was a fashion, a titanium hype or a ceramic hype. Companies tried to do or to produce prostheses just because they were so popular, the materials, and they were in reality not really the best materials for the use. This is a table which shows you that even within the basic materials like titanium, cobalt chromium, polyethylene, there are a number of undergroups like wrought, cast, and so on. And this is one table where we got approval in a specific regulatory jurisdiction. Now, in general, titanium and their alloys are excellent for cementless hip stems and direct bone contact components, but not as good for wear partners. Cobalt chrome alloys are excellent for cemented stems and are, if highly polished, good wear partners for poly. Aluminum oxide ceramics are excellent wear partners for poly or ceramic counterparts. And the poly, again, is divided in traditional poly, highly cross-linked poly, and alpha tocopherol or vitamin E doped, which was much later introduced, but which is nowadays a very popular poly because the vitamin E grabs the free radicals, the oxygen, and at least prolongs the life, to be careful in statements, prolongs the life of the poly. Oh, come on. Now, but these are not iron rules. Are titanium stems only non-cemented? This is the general rule. Titanium, hips from titanium, non-cemented. Hips from cobalt chromium or stainless steel, cemented. Basically, yes, but it depends. When we look at the young modulus, cortical bone, 20 gigapascal, cement 2 gigapascal, titanium 110, cobalt chromium 230. The rule is that the flexural stiffness of a system is defined mechanically as the product of Young's modulus and the geometrical moment of inertia of the diameter around the bending axis. What does that mean? Sounds very complicated, but you see a cylindrical rod of 10 millimeters. Titanium E module 110 gigapascal. We load it with 1000 Newton, and the maximal deformation is 6 millimeters, like you see here. 6 millimeter deformation with a load of 1000 Newton. E module 110. Now, when we do the same thing with the same rod, 10 millimeter, with cobalt chromium, you see that the deformation is only half of it, 3.5 millimeters. Same load, same size, but 3.5 millimeter. But now we increase the size of the titanium rod only by 19%. Still 110 gigapascal, still 1000 Newton, and the deformation is the same as the cobalt chromium rod. So in other words, if the size is right, you can also use titanium for cemented stems. That is something many surgeons, when we do it as a custom made, for instance, and we provide titanium stems for cementing, the surgeon tells us, no, titanium cannot be cemented. Wrong. If the size is good, you can cement it, but the size has to be fit to the necessity to protect the cement. So let's remember the flexural stiffness of a system is defined mechanically as a product of Young's modulus and the geometrical moment. This means that an increase in diameter of about 19% is already sufficient to achieve the same flexural stiffness in a titanium stem as in a slightly thinner cobalt chromium stem. Stem thickness is important. This is also very important to tell the surgeon. There are always surgeons, some surgeons had a bad breakfast maybe, here's a patient, now I use a thin stem that's easier to introduce. And later there is a loosening. You need the largest possible stem intramedullary to protect and to give a good support for the cement layer. Now, what do we do when we produce a new knee system? We do range of motion analysis, word test, particle analysis, compatibility test, roughness measurement, minimum thickness measurement, and of course, biocompatibility. Here, a view how these tests are done. There are not only 1 or 2 or 3 machines. No, 6 pieces have to be tested to give a statistical acceptable answer. A snapshot on a test for a new knee. Actually, I asked my engineers, how many tests did we do to develop this new LSK knee system? And they showed me the tests, 32 comprehensive tests, including implant design test, instrument test, usability and coding test. So 32 tests, that is quite a lot, but it is necessary to be sure to have a good feeling. For instance, when we have when we test a new stem, we don't do only the ISO 72064, the test which is necessary for a hip stem, but I always want to see the so-called Locati runout test. The Locati test means we step up with our testing until the implant fractures. From that, I get the information, how strong is that? Is it only a little bit about the requirement, or is it a long way about the requirement? I want to know that. Now, something new technology, 3D printing. Everybody talks about it. Our machine That's what you can do with 3D printing, little trees and towers and everything. And this is one of the very large pelvis replacement, which we did for a patient, I think, in Romania, just as an example. Now, how do I get my 3D printing approved? And that is a little bit a problem. If we have a regulatory authority which goes only by standards, I really have a problem. There are no standards, but there are technical standards within the which are not official ISO or ASTM standards, but which are standards from knowledge, from history. That's why I put here, don't try to reinvent the wheel. So when we look at a new development, a new material, first we should look at the chemical purity. Second, at the physical values. Third, are the same mechanical strengths inherited? And if those things fit, you can already be quite sure that you have a product which will work properly. Of course, difference in product design must consider complementary approaches. 3D print requires hip treatment. 3D print must include the parallel production of tensile bars. So we check every batch, does it have the correct physical properties? And of course, if new structures are produced with a new technology like 3D printing, cleaning procedures have to be found and tested in order to eliminate the debris remainder from the powder application. This 3D printing, you probably all know it, that's a powder, usually titanium, could also be cobalt chromium. It comes down like a little bit of rain, and then comes the electron beam and melts it together to the shape of the product. But a lot of this powder stays within the product. And we have to be sure to get these debris out of the product. Otherwise, can you imagine when the patient has such a product in his hip or in his knee, and a little piece of titanium comes into the sliding area? This would be catastrophic. So what do we do? How do we go about? We at Link approach this with view of the known procedures to clean similar rough surfaces like plasma spray. And then we compare our results with already approved products. Now, lower prices, same quality. Excessive price deterioration, exaggerate pressure for price reduction can also do some bad things on implants, on products. We have experienced that. We know this from some markets where low price products are in the market. Like similar or comparable, couple decades ago in the automotive industry, they reduced the quality and the cars were of no good quality, but the customers pushed the manufacturers back to a high quality. Of course, it has to reflect in the prices. Or unavailability of necessary products. If the prices are too low, we see this in Germany now in the present pharma market. Some pharma products, especially for children, which have low prices due to the pressure of the social systems, went down. What did the pharmaceutical companies do? They look, where can we get it? From India, from China. Okay, and now, you know all, we have a special political situation in the world. The supply stopped. Now we don't have these products in Germany. This can also happen with medical technical products if the prices are not adequate. There are lots of ways to reduce production costs that might lower quality. And that's what we don't want. And I just give you a couple examples here. This is, this is of course a competitor knee. And you can see, when you see the large picture, that this knee still has a cast skin. You can see it by this line, because that is where the two parts of the die came together. When you see this on a raw part, you can be sure the cast skin is still present. We at Link, we reduce 100% all the cast skin. This is the rule, even in areas which are not prone to fracture. Or in a very small number of poly parts, you can find extraneous matter particles that can only be detected under strong through light. These particles are rare, but should not go in circulation. Here is an enlargement. You see this particle. It's not on the surface. It's somewhere in the middle of the product. And we see it with a strong through light. And this, of course, is scrap. Now, back to the regulative challenges. NDR regulation has not improved patients' protection to our belief very much. A number of rarity product lines have been discontinued. Manufacturers may be hesitant to enter markets with too high demanding entry burden. New product development are driven to markets where regulatory approval and achievable prices are in a well-balanced relationship. And this is also a big problem in regulatory. Some of the regulatory authorities, either they are unsure about a thing or they want to be popular and show that they protect the patients even more, like it happened in Europe. They increase the bureaucracy. And the increase of the bureaucracy does not improve the products. Of course, it may avoid this thing. There was a criminal act with the French PIP breast prosthesis, where they use cheap industrial silicone. This could be reduced, but in general, the bureaucracy does not improve the quality. Now, here we have an overview of the regulatory requirements, the authorities in the EU, notified bodies, government authority, in the USA, government authority, and in Brazil, government authority. And the registration requirement, we see here, different papers, different rules for registration requirement. Contact the authority, free choice of notified body in any EU country. So we could go to any notified body, but of course we have our special notified bodies that know us, that audit us. So we have a certain relationship, and that I think makes much sense. It gives a better understanding. We can have discussions about implementation of requirements which are possible. FDA, language English, meeting can be scheduled, discussion about implementation of requirements is possible. Here we have a little bit of problem with Anvisa. Language Portuguese. Internationally, it is English. Meetings can be scheduled, but only in Portuguese. Of course, we have people who work here who speak Portuguese, but if Anvisa wants to comply a little bit with the international rules, with the international requirements, they should also work in English. And discussion about implementation of requirements are, of course, possible, but again, we have to use Portuguese. Now, specific requirements here in the EU, official language of each country, but we can use English. Also, we have 26 countries with different 26 languages in the EU. USA, only English language, and Brazil, Portuguese language, registration number and label. Timeline for registration currently at least 12 to 18 months. This is terrible. They are loaded with requirements. They are loaded in the EU with new applications, so 12 to 18 months. USA, very nice, very strict timeline for response. Within 3 months, the US FDA has to answer at least, not give an approval, but they have to answer our submission. So we get an answer and then we can go from there. And now I learned that in Brazil, you also have such a rule. Whether it's always fulfilled, I'm not sure, but you have a strict, you have a rule where you should get the answer. Now, publicly available information, the regulation available in several languages in the EU, the guidance document mainly in English. In the US, regulation guidance document and training documents available in English. And in Brazil, regulation in Portuguese, not easy to find for a foreign manufacturer. Recognized standards in the EU, most ISO and ASTM, harmonized standards and common specification in the US, most ISO and ASTM, and also in Brazil, most ISO and ASTM are recognized. So what can you do to adequately control implant quality and best protect the patient? Use existing technical standard rules, not necessarily ISO ASTM if we are talking about new technologies, but then discuss between the engineers what is technical standard in daily use. Control development and manufacturing and test documentation, regularly audit the implant manufacturers, check product labels, marketing materials, and user manuals. Establish a national register, very important. The Swedish register and the European registers has shown and pointed out which products work very well and which product might have a problem. Set up an EndoCert program to certify hospital surgeons according to latest scientific data and specialty centers for joint replacement. This is also very important because the implant is at the most 60% of the deal in a joint replacement. At least half is what the surgeon does, his knowledge and his ability. And this we do in Germany with the EndoCert program. Hospitals are certified, they have undergone a special training, and this gives a certain security. So, what not helpful is regular product recertification of established grandfather products. This is something which we have to do now in the EU. Products which are a long time on the market, we have to recertify. Request for clinical data for established products, like recertifying aspirin. Does it make sense? Full certification for marginal products, sidelines, custom products. Increased bureaucratic workload doesn't bring us any further, as I said before. So in summary, medical device submissions in Brazil are not easy to handle. Good communication with distributors is essential for success. Ideas for improvement, acceptance of complete implant system submissions, more publicly available information, meetings in Portuguese and English. Proud to be part of the medical device market in Brazil. Looking forward to further submissions. Thank you very much.
Parabéns, Dr. Helmut.
Congratulations, Dr. Helmut Link, for your presentation. Do you hear me? Can you hear the English translation? Do you hear the English translation? Yes? Testing, testing. This is the English channel. 1, 2, 3. Can you hear the English translation? Testing, testing. 1, 2, 3. 1, 2, 3. This is the English channel. Do you hear me? Yes? Okay, perfect. Thank you. Excellent. All right, so I'm going to speak in Portuguese. Right, Dr. Helmut Link, congratulations on your presentation. I believe that for us here in Brazil and at this event, it's such an honor and privilege to have you here doing this presentation about orthopedic implants and about all the requirements and what the company Valdemar Link and the group does to meet all the safety and efficacy requirements. Also, you know, the challenges that there are in the world, because each regulator, besides having common requirements, they also have specific requirements. So this is a challenge for all companies in general. And so your experience and your sharing of implant development really enriches our conversation. Considering that scenario and all the challenges that we have, we know that orthopedic implants, and the majority of them, are based on technical standards for raw materials and processes. And you have mentioned the challenge for non-standardized implants, and we have that for several types of implants. So thinking from the standpoint of regulators, because we receive some criticism about, you know, their accepting our submissions, I would like to ask you the following question so that we can learn from you, because we're here really to, you know, share knowledge and learn. So what would be your suggestion regarding the submission of documents to actually prove to regulatory authorities and Anvisa these cases of non-standardized orthopedic implants? We know that a lot of protocols are created by the company itself. So what do you think would be something that the agencies could do to make this comproval of efficacy and safety standards easier?
Thank you. First of all, I hope I'm not understood. Whenever there is a standard, use a standard. I only point at developments or at products where standards are not applicable. And a good example is this 3D printing. Now, what I showed in one of my slides is, what do we have to look at? Is the material biocompatible? We don't really, we can see this also in standards. So if the 3D printing material complies with the biocompatibility standard, fine. But now we come to the physical strengths of the material. That could be different. So if we could prove, we as a manufacturer or another manufacturer could prove that the mechanical data fits or compares to wrought titanium or to casted titanium acceptable, then also it is not written in a standard, this should be accepted from that view. So that's what I mean. Yeah, that's what I mean. The engineers know about physical strengths and about surfaces and about these things. And when they have a new production technique and they view step by step every parameter, and the parameter is similar to what has already been done with a standardized material, the solution must be, it's okay.
Não, tá funcionando? Tá, né? Então, só
Right.
para esclarecer, a gente tem.
I just wanted to clarify that we have been accepting in the case of Anvisa. I think the biggest challenge that we have, based on what we see, you know, based on what you presented and everything, that a lot is done in the plant. And so the challenge is to kind of transcribe what's done in the manufacturer so that we can present that in a document. Because based on our experience, we know that there is a lot of care regarding the manufacturing of medical devices. And maybe, you know, the translation of that quality language that is used in the manufacturer is difficult, you know, to translate that to the regulatory authorities. And so the goal is actually, like you said, to present something that has quality for the final user. So to complement those challenges, and based on your resume, you showed us the experience in 3D printing. So what do you think is the biggest challenge regarding the manufacturing of implants through 3D printing? Is it the process? Is it the raw material used? How to prove, especially considering customized products that sometimes, you know, are made, there's only just one single part that is produced, and some of the tests are not conducted because it's not viable. So how would be the whole process, and what could we do to offer like a solution or a suggestion? What is your take on that?
You have to know the procedures. You have to learn how the procedures work. And the one point you mentioned, what is the biggest challenge in 3D printing? It's, to my belief, it's a cleaning, to get out the debris. And that's why we try to go parallel to our experience, for instance, with plasma spray. Plasma spray is also something where particles are brought on a surface, and where you have pores where these particles can hide. So that's the same thing with the 3D printing. And when you compare, and then when you go deeper into this and count, and maybe even what the Americans did, they asked us, compare your 3D printed component with a competitor component. We did that, which was already approved in the US. And we have the same counting of particles. The resolution is, it's okay.
Certo. Eu não recebi aqui nenhuma pergunta.
Excellent. We did not receive any questions through the app. Let me see if we have questions from the audience.
Não?
Any questions?
Eu acho que precisa ser no microfone.
We need you to speak in the microphone. Good afternoon. So the process of receiving purchase materials to 3D printing follows the same purchase processes for materials purchased in blocks or in bars.
No, of course not. That is a different type of material. A block or a bar can be checked differently than the 3D powder. Here you have to audit a special supplier. You have to check whether this goes along with the requirements the machine manufacturer tells you, and where you can be sure that you can produce the parts you want to have with this material. And then you have to stay with that supplier. And of course, from time to time, you have to do these checks again. But it's more difficult with the 3D material than with bars or blocks or something like that.
Alguém mais? Acho que não.
Any other questions? So Dr. Helmut Link, thank you so much again for your presentation and for answering our questions.
The next meeting that is held with us, we are going to speak in English with you.
And I have a question to you. Can we in the next meeting maybe also talk in English?
For sure. It was what I was saying to you. We are going to speak in
Very-
English the next meeting.
Very good. Thank you
For
very
sure.
much.
Thank you.
Agradecemos a conversa e os esclarecimentos do Sr. Helmut Link e da Priscila.
I'd like to thank Helmut Link and Priscilla Consigliero for their participation. So now moving on to our last panel, which talks about the validation of tests for coded implants. So I would like to invite Marcia Ribeiro from the Coordination of Implanted Materials at Anvisa. And we would also like to invite on the stage Mr. Patrick Aldinger, Senior Manager of Hip Development Products from Smith Nephew in the USA. Thank you.
Ok, vamos lá. Só iniciando aí a
All right, so to start our presentation, welcome, Patrick Aldinger. Thank you for accepting our invitation to participate at this forum. This is such an important moment for us. This is a moment where the sector has more involvement with the industry, and this allows us to exchange experiences because we do not know everything. And so I understand this moment is really for that, for us to, seek improvements on both sides. So Mr. Patrick Aldinger graduated in 2001. He's a Master of Business. I'm sorry, I do not have my glasses on. One sec. Master of Business Administration in 2006. He's with Smith Nephew for 20 years. He started as a research engineer and is currently Senior Manager of Tests, Research and Development. Patrick Elniger, the floor is yours. I'm not going to make any, as difficult questions.
Thank you. And thanks to the organizers of this event for inviting me. And thank you for just being patient with me. I had some travel issues, so I didn't get here till today. So, and again, thank you for sticking around for this I think this is the last talk of the day. And to talk about porous coatings, specifically what we do as an industry, as an international orthopedic company to evaluate porous coatings used in various different implants for reconstructive devices. She covered my background. So yeah, I've been in the industry in R&D for over 20 years, in R&D doing testing of various different types. Over those 20 years, I gained a lot of experience with finite element analysis, corrosion testing, mechanical testing in general, and retrieval analysis. And currently I'll work in our product support group, which is mainly like a sustaining engineering type role, managing legacy devices. So both for hip development and for testing. So what I'd like to cover today is the history of porous coatings, some of the industry requirements that we look at as industry to evaluate coatings. A lot of the test methods, you'll see a lot of ASTM standards, ISO standards. I'll go through most of them. The acceptance criteria that are defined in some of these guidance documents and standards. And then kind of round the discussion with a little bit of clinical performance and then some closing statements. So in regards to the history. So coatings were developed with the intent of improving implant fixation. I realized when I was giving this talk and I heard Professor Helmut get up here, he was talking about other coatings. I'm particularly talking about porous coatings used for implant fixation. I'm not talking about surface coatings, so just to be clear. They were first developed in the early 1900s for use in dental applications. Specifically for artificial tooth roots. And in fact, in the early 1900s, the original devices were patented. There's a metallic-like cage for that same application in the tooth root. And then for the next several decades to the 40s, there was a lot of tests that went on to evaluate different type of coating technologies and applications. And in the 50s, Brainmark demonstrated good osseointegration with a tooth-like implant. In addition to this, in the 1960s, they started to understand that there was a lot of deficiencies with cemented type devices. And so because of that, porous coatings started to get even more interest for use in orthopedics. In the mid-60s, it was first used in dental practice, and then in the late 60s, the first porous metal implant. And then again, as you're getting into 1970, there's a clinical trial with a stainless steel hip stem that used a flame sprayed coating. It did not do very well. It had issues with corrosion. But it was a kind of a first attempt at a clinical trial with a porous coating. In the 60s and 70s, Loic and Galante developed a titanium alloy mesh. And then as the 70s roll on, even more animal studies and more studies on different type of coating technologies are existing. And so then you roll into the 1980s, and that's kind of a big push for porous coatings being used in hips, knees, ankles. And then fast forward to even later up to now where we're looking at 3D printed structures. Some of the first ones introduced by Lima and Adler. An image of a couple of different types, a porous coated centered bead stem at the top corner, an ankle device at the bottom, and then a tibia base plate, as well as a 3D printed acetabular shell. In addition to just traditional porous coatings, there's also hydroxyapatite, which is a coating that's applied either directly to a stem or on top of another porous coating. And so its first use was about in the 1950s as a bioceramic bone defect filler. And then in the 1980s, as centered bead technologies and plasma spray become more widely used, HA is now being used placed on top of it to promote good bone ingrowth. And so you have this term now of cementless or press fit implant technology as compared to cemented implants. So I'm sure there's other coatings that are out there, but I'm going to cover the most well-known ones. And so typically it's going to be two materials, cobalt chrome and titanium, or a titanium alloy. You have cobalt chrome and commercially pure titanium and titanium alloy centered beads. And then you have commercially pure asymmetric powder. And then you also have a fiber metal. So the typical process for applying porous coatings, at least with sintered beads or with beads, is sintering. And this is a high temperature process for the material or for the entire implant, including the pore structure. Plasma spray is a little bit different. It's not a high temperature process for the substrate material. It is a high temperature for the powder itself. It's being sputtered onto the surface. And then HA can also be applied in the same manner or a similar manner. And then also 3D printing, additive manufacturing using electron beam or laser fusion. So an image of a couple of different types of systems there, a hip stem with sintered beads in the top left, a fiber metal mesh on the top right. We have just plasma spray titanium on a Ti-64 stem in the lower left, and then an HA coated over a roughened titanium stem. So right now, the current applications for porous coatings is hips, knees, shoulders, and ankles. So just a nice little cross view of what one looks like. So the bottom is going to be your substrate material. As you introduce this into a high temperature environment, the beads melt into the substrate. Now these beads are generally applied to the stem with some sort of binder, like a glue that just holds the beads in place until they cook it in the oven, and it fuses to the substrate, fuses to each other. This is also an example of an HA-coated sintered bead structure. Asymmetric powder, which was introduced a little bit later from porous beads, is using titanium powder, using a very similar technology of sintering it, putting it into an oven, letting the substrate fuse with the powder, and then the powder fuses to itself. This is a cross-section I pulled out of a book of various different porous coatings. We already kind of covered cobalt and titanium beads, spherical beads up at the top, asymmetric powder. This is also a powder that's also, the powder itself was also porous, and then a tantalum foam. Here's an example of a 3D printed pore structure. So in this particular case, the substrate was printed as a solid structure, and then the porous coating itself was built upon that. And with 3D printing, you can adapt any kind of structure you want to apply. You can be heavily reticulated. It could be just struts. That's the beauty of 3D printing, is you can adapt it to whatever shape and size you want. Most likely people are trying to base it off a cancellous bone structure or trabecular bone structure. So some of the early challenges with press fit technologies, at least very early on, was there had to be precise implant matching to the canal. So with cement, you could create a canal and put the stem in there, and the cement, you'd have a cement mantle that was a couple millimeters thick, and it didn't have to be completely nicely conforming. With press fit, it had to be a much better contact with the bone, so you had to have really good broaches. With early press fit designs, there was notes of early stage pain. There were challenges with tubal components specifically. Stress shielding due to modulus mismatch that Professor talked about, trying to optimize that moduli with the bone, and using cobalt chrome stems with cobalt chrome beads for long cylindrical stems. Those had issues with stress shielding. Metal ion release. So early technology with porous coating, some of these, they could flake off, that could get introduced into the joint, and that could be problematic for wear. And then just in general, the loading mechanisms across hips, knees, ankles, and shoulders is different. So trying to take what was learned for hips and knees and just shoving it into hips or into shoulders and ankles may not be the best thing all the time if it wasn't totally understood. So there were just differences at the time. So to look at usage. So I pulled this from the Australian registry. This is the most recent one. So over the past couple of decades, cemented use for hip stems has kind of decreased, whereas cementless stems have increased slightly over the past several years for sure. And then hybrid is kind of in the middle, where hybrid is you have one component that's cemented and the other one is not. For knees, it's a little bit different story. So you have a high usage and continuing to be high usage, although it is declining now for cemented knees, with cementless also starting to increase, and then the hybrid version also starting to increase in the last couple of years. Could be due to 3D printing. A lot of device manufacturers are coming out with 3D printing on knee components, and they're performing much better. So that could be a reason why we're seeing an uptick now. For shoulders, so this is an interesting graph because with shoulders, it's a little bit different. I talked about the loading mechanisms are a little bit different. The primary use for right now for shoulders is with the glenoid being cemented, and the other parts can be cementless. And you see a decline in cementless only over the last couple of years, and then a slight decline on cemented only use. And partly this was captured in the Australian registry. They mentioned that to get lower rates of revision using cross-linked polyethylene and cementing the glenoid had better results. But in the ankle, it's a little bit not as understood. There's not as many ankle implants going in. There is a difference in the way the FDA views ankles versus internationally. So that could be part of the problem, but we just don't have enough data. So industry requirements, what do we look at as a device manufacturer to guide us through how we evaluate coatings? What we have are mainly FDA guidance and ISO standards. And so, for instance, the FDA guidance document for testing orthopedic implants with modified metallic surfaces opposing bone or bone cement, and then a general implants coating for coming out of ISO. I think it's important to note here that this guidance document came out in 1994. As I mentioned, orthopedics, the use of porous coatings in orthopedics kind of really started to become prominent in the 80s. So for a while there was no guidance, and then there was pretty early on, 1994. This is still an active guidance document that's used today. Within ISO, there are even more guidance related to HA and plasma spray. In ASTM, there are several standard specifications for knees, hips, shoulders, and ankles that include a lot of the same information from some of the other guidance documents. So right now we have 3 different large groups of standards and guidance documents that kind of guide us in how we should be evaluating porous coatings. So part of the requirements are the metallurgical side, the microstructure of the pore structure itself. And so from a metallurgical side, how is it affecting the substrate material? What is it composed of? What is the microstructure of the porous coating you're actually using? The thickness, the location, shape, size, something called mean void intercept length, or MVIL, and porosity. Those are all requirements from the guidance document. And I'm not going to talk about those. I'm going to be talking about the other performance tests that we do. So just be aware that these are other requirements within those guidance documents and international standards. So in terms of testing, what do we test when we look at porous coatings? It's pretty much the same story for both the guidance document and ISO. There's essentially 4 main tests. There's shear fatigue, static shear, static tensile, and abrasion. And as I mentioned earlier, the FDA guidance document came out in 1994. Shear fatigue wasn't listed with the ASTM test standard that exists for shear fatigue, and that could be due to it when it was developed, when that ASTM standard was developed. Same thing with abrasion. But nevertheless, it's the same 4 types of tests that are being done. And one of the other things that the guidance document mentions is, the FDA guidance document mentions, is the mechanical properties of the substrate. Because we can look at the porous coating by doing some tests that I'll show in a minute, but how does it affect the substrate? I mentioned previously that there's characterization that needs to be done about the substrate, but this is different. This is how does the strength change for the substrate material? Because as I mentioned, with beads and asymmetric powder that use a sintering process, it does affect the microstructure of the material. So how does that affect the long-term performance of a hip stem or a knee component, whatever you end up making out of? Maybe the pore structure itself is fine, but now you've weakened the stem. So the guidance document asks you to review those type of scenarios. Okay, so the different test methods, I listed I think all the test methods that exist for evaluating porous coatings, and I'm going to kind of step through each of them. But before I do that, just to give you an idea, most of those test methods, actually all those test methods that deal with the four main components of static and shear fatigue testing, tensile attachment, and abrasion, is all based on a coupon. So these are all going to be a cylindrical coupon that's flat, that has the porous coating on one side and the substrate material that you will be using eventually for whatever implant you're going to make. It goes through the entire manufacturing process that you will use for your final device. Same thing with the HA that can also be applied to the porous coating. But these are all going to be coupon-based tests. And in order to evaluate the mechanical properties of the substrate, you can do coupon-based tests. There is a tensile or a rotating beam type test you can do on a coupon that has gone through the full manufacturing process. Or you could even evaluate a hip stem, like was shown earlier with the ISO 726-4. That's a test method for evaluating the distal strength of a hip stem. You could do the same test, and that will help you evaluate how strong the stem is that has gone through the full manufacturing process of a porous coating. So a key part, because this is a coupon-based test, is sample preparation, which is very key for these types of tests. It needs to go through the entire process. It needs to be clean like you would for the final device. The way these are prepared is you have your coupon, and one side has the porous coating on it, and then you have a test stub, in this case on either side, that is compressed with a thin film that's an epoxy or some sort of adhesive agent that sits in between that cures the porous coating to the stub. And I'll show the test sample in a minute. But each of the ASTM standards calls out that these epoxies or adhesives should have a minimum strength, or they should be as strong as the required strength of the coating. Because one of the standards, I think it's actually all of them, I think call out this 34.5 MPa. If the strength of the coating doesn't necessarily have to be that high, I don't know if the adhesive strength of whatever you're going to use there has to be that high. But nevertheless, this is what is called out in the standard. These call out using FM 1000. There are some other options that are listed in some of the other standards. But essentially, you need some way to cure or adhere your porous coating to a sample so that you can try to break apart the porous coating, because that's the whole point of these tests, is to see how strong the coating itself or the coating interface to the substrate is. So just, you would place this whole thing into an oven, and FM 1000 in particular is heat sensitive or heat cured adhesive. So you put this into an oven at 350 degrees Fahrenheit, and it'll fuse into the porous coating. Sometimes you apply a dead weight, or you could also do a spring-loaded setup. What's key about when you do this, I don't know if it's called out in the standards, is taking one of the samples after it's gone through this curing process, and it's basically destroyed, because you want to cut away all the adhesive until you get to the porous coating and make sure that adhesive hasn't gone into the substrate. Because if it does, then you're not accurately testing the porous coating. So the bonding agent is pretty critical for this setup. And these setups, there's not a uniform weight that will just always give you the exact same amount. So that's why you take that one coupon and you destroy it to make sure you haven't gone too far with the penetration of that glue. So this is just try to get a close up. It's kind of blurry, but this is the side with the porous coating, and then your test stub on one side. So ASTM F1044, this is static shear testing. So in this case, you're applying a tensile force across the sample at 2.5 millimeters per minute until failure of the coating. In this setup, it's important to note that there's a universal joint or a multi-directional joint here, so that you can always apply that axial force and you don't get any kind of side loading off that coupon. Because most of these coupons, as you can see here with this particular setup, is 19 millimeters in diameter, so it's pretty small. But if you only load one side of it, then that's not really telling you enough about the strength of that coating. So you have to have a setup that does allow you to pull this perfect as possible in axial tension. So in terms of the sample size, so ISO 13779-4, which is for HA, calls out 10 samples. What's called out in the FDA guidance document is kind of a blanket statement about the sample size for all the 4 tests that I mentioned, that you should just use 5 samples. In previous clear devices, they've also found, the FDA has found a sample size of 5 to be acceptable. Shear fatigue is basically the same thing, except now this test construct is in, excuse me, a fatigue load. You can apply this up to 30 hertz, and typically this test runs for 10 million cycles. Again, sample size is 5. In ISO 13179-1, it says 5 as a sample size, as well as I mentioned before, the FDA guidance also calls out 5. That is the same coupon size as the previous static shear test. Tensile attachment testing according to F1147. So this is a tensile test pulling apart the coupon at 2.5 millimeters until failure. This is not shear. This is the surface, the entire surface being pulled in axial tension. Again, based on that ISO document for HA, it says 10, but the FDA guidance document calls out 5. This is a bigger coupon. This is a 1-inch in diameter coupon. But the same care is taken in making sure that you apply the load as perfectly in axis as possible. So what is a failure in these tests? So in ISO 13779-4, it does call out the different types of failure modes you can see in these tests. And so going back to the image I had earlier, I actually flipped it. This is the substrate at the top. Here's that first bead fused to the substrate, and then all the fusion, all the beads being fused together in the HA. When you apply this film to the substrate or to the surface of the porous coating, just imagine this is your epoxy. So you can get a failure in the interface of the substrate and the bead. That would be a failure type A, according to that particular ISO spec. You can have a failure in the coating itself. So it could break here at this bond, or these bonds where the beads are fused together, or asymmetric powder, whatever the porous coating that you use. It can be between the interface of the coating and the adhesive. So that thin layer of bonding that in this case would be adjacent to the HA coating. And then finally, the adhesive itself fails. Or you could have a mixed mode where you see a variation of all those. And that's where how well you align that test sample could have a big influence on what type of mode you see. And according to this ISO spec 13779-4, they call A and B a valid failure mode, and C and D are invalid for HA, because this is specific to HA, the standard, meaning that if you saw that failure mode, you would have to toss that sample aside. And then E is valid only if you see a mixture of A and B. So taking care of what the samples look like when you test them and visualizing them after the testing gives you a lot of insight. Did you have a valid sample for the test? And this is just an image of a bunch of different types of failure modes. I'd say this is one where you pull the porous coating directly off the substrate. And this one is more of a adhesive to coating failure. You do see a little bit of coating. I mean, adhesive adhered still to the top is the coated surface. You do still see a little bit of adhesive adhered to the porous coating. This almost appears to be fully just an adhesive pulling off of the porous coating, but you can also have mixed, right? And so in some of these mixed modes, it's kind of hard to tell. It would depend on what the strength was. And eventually I'm going to get to the acceptance criteria that we've established. You know, if you do a test like this, and as long as you ensure that your adhesive hasn't penetrated to the substrate, you do that when you prepare the samples and make sure that your setup was good. After you review your failure modes, if those loads are high enough, it seems like to me you could still have some of the other variations of failure modes and it still be an acceptable test. So those are all the test setups for those first three that deal with static shear, fatigue, and tensile attachment. There's another one that was a requirement in the ISO guidance or in the ISO document and is kind of spelled out a little bit in the FDA guidance document. It's called Tabor abrasion. There's a machine called a Tabor abraser, and you put this big cylindrical shaped coupon in there, and it's almost like an old school record player. It just kind of spins around, and there's two little wheels that rub against it and break off the coating. So according to that standard itself, F1978, you test 6 samples. And what you would do is you would weigh this coupon before you do the test, and you can weigh it after, or there's a way you can just vacuum up the porous beads that are being sheared off as you're running it through this test. So I mentioned, the FDA guidance document mentions that you should evaluate the substrate material that has gone through the full manufacturing process that the sintering would go through. And typically you'd want to do that with the beads on there, because in the case of fatigue testing of hip stems, you want the beaded surface on there because that could create stress concentration areas for the stem. But again, you can do this based on a coupon level type test. Still, it's a sample that has gone through the full manufacturing process. You can do hip stem testing where you could cause it to fail in the porous coating, and it could tell you the strength of the substrate material and the coating. You could also use something like this, which is ASTM F1800, which is the unsupported tibial base plate fatigue test. Now, this test is not putting the porous coating in any kind of stressed environment, but you're still testing the device that has gone through the manufacturing process. So it does give you insight as to what the porous coating might have done in terms of strength to the base plate. So in regards to worst case, so really the only guidance that we have is in ISO 13779-4. And what it mentions is essentially that the thicker the coating, the more worst case it's going to be. And even so much so that you can make it thicker than what your specification really calls out, if you can ensure that that doesn't somehow, give you the wrong answer. But it's basically saying the thicker, the better. And you think about when you do this test, you're having to cure this stuff together with an adhesive film. It's nice to have a little bit of room there. So, but it depends on what your specification is for how thick of a coating do you want to use. People use multiple layers of beads, like 3 layers or maybe 2 layers. Asymmetric powder, it gets a little harder to say what the layers are. It's more of a thickness. Same thing with some of the other technologies that are out there. It's more of a thickness. So typically for a worst case setup, you're wanting to use the thicker or thickest coating, and sometimes even beyond that. So the acceptance criteria. So what do we rely on as in terms of acceptance? So in the FDA guidance document, as well as some of those ASTM standard specifications that I mentioned earlier, they do call out what is a load that is deemed acceptable. So for the FDA guidance document in regards to static shear, it's greater than 20 MPa. This is spelled out on page 4 of the FDA guidance document. Both the knee and the hip standard specifications state the same thing. ISO also calls it out in 13179-1, and then the ankle standard spec also calls it out. For shear fatigue, there's not really a load that's or a force that's been deemed acceptable. It's the only real acceptance criteria is that it is tested to 10 million cycles. So in this regard, you could test a comparison device, a predicate device, and use that as your minimum strength and compare your new coating to it. Tensile attachment, kind of that same thing of 20 MPa in the guidance document in both the knee and the hip standard specifications from ASTM. ISO is a little bit different. It calls out greater than 22 MPa in that particular standard in that section. For abrasion testing, the FDA guidance document doesn't call out anything, but the ISO spec 13179-1 does call out that the loss of material that you see in that test should be less than 65 milligrams after 100 cycles. Specific to the HA coating, The static shear strength should be greater than 15 MPa with no samples falling below 10 MPa. And that's called out in that particular spec in that section. The ankle states the same thing. For shear fatigue testing, it's again greater than 10 million or greater than 10 MPa for 10 million cycles. So in this case, for shear and HA coating, it does call out a requirement for strength. But this isn't an annex, so it's considered to be optional. Just make note of that. For tensile attachment, it doesn't specify, and abrasion, it doesn't specify. So again, in that regard, you could compare back to a previous technology, or you could fall back to possibly one of the other tensile attachment strength values if you want to. Okay, clinical performance. So the way in which we tested these devices over probably the last 30, 40 years, well, at least since the FDA guidance document that came out in 1994, was to do this coupon-based approach to evaluate what the values are, to evaluate the strength of the coatings and meet those specifications that are called out in that guidance document. And from that, we have determined what is acceptable. So these stems that have been out in the market for decades that have those technologies on them have performed well clinically, which I'll get to in a minute. But kind of the approach that was taken was these coatings were evaluated first as spherical beads were kind of the first ones to come out. Then it became titanium powder or asymmetric powder, and then now we're getting into 3D printing. So that was the way in which all these technologies were first kind of are being developed now and were developed over the last several decades. And it seems to be an approach that has worked well because press fit technologies are working really well. And most of the device manufacturers adhere to that guidance document. But the FDA guidance document does require that if there is any clinical data, they need to see it. So if you have data on your coating, you need to submit it. So if you're coming out with, let's say, a new hip stem design, but you're using asymmetric powder, they want to see that clinical data. It's for a new application, it's a new hip stem, but they would still want to see that clinical data. So just kind of round it out. And now I only have our Smith & Nephew's devices here to show. So I pulled a couple that I could clearly see what the complaint rate was based on our own internal CERs or clinical evaluation reports. And so what has been stated through clinical literature is this particular acetabulum design, which is called R3. It is a shell with stick tight, which is an asymmetric powder. And that powder can also come in a form with HA over the outside of it. So its survival rate is 97 to 100% from 3 to 7 years, with any reason being noted as the endpoint. So that could include anything that ever happened, not necessarily something that was a result of the porous coating not working. So that's why I wanted to pull this other information is we had 55 complaints out of about 1 million that were reported to be due to loosening, implant migration, or osseointegration problems that could be associated with the porous coating. It's not saying that's what happened, but they could be. And that was a rate of 0.005%. A polar stem, which is a plasma titanium HA stem. So plasma spray titanium over the surface of the titanium alloy and then HA over the top of that. So its survival rate is 99.7% at 3 years and 99.6% at 5 years, and even at that same rate at 6 years. And again, looking at the complaint side of it, 12 complaints out of roughly 376,000 due to loosening, implant migration, or pain, which is a retrieval rate of 0.003%. And then another one, this is Anthology, which uses the same asymmetric powder coating that the R3 shell has, so just showing it in a different implant application. Its survivorship is 96 to 100% at 4 to 10 years, but when you're looking at just the stem itself, it's 98.5 to 100% during that same time frame of 4 to 10 years. So really isolating the stem itself, which was a asymmetric powder coated stem. So in this case, there's 31 complaints out of roughly 260,000 due to osseointegration problems, loosening, implant migration, or pain. And that's a retrieval rate of 0.012%. So just to kind of summarize a little bit, the way that we have evaluated coatings over the last 30 or 40 years has been using this coupon-based approach of looking at tensile attachment, static and shear fatigue, and abrasion testing. Utilizing these FDA guidance documents, ISO standards, and the ASTM guidance, the standard specifications, as well as their test methods, has resulted in us producing successful porous coatings as an industry, not just a Smith & Nephew, as an industry. So just to kind of end on what is the future of coatings, Professor Link mentioned a little bit about 3D printing and just how it is really paving a new road for porous coatings. You can do so much new things with it. As I mentioned on the knee side of it, now you can produce coatings on knees that perform better than the original historical ones that came out that didn't perform very well. They had really large beads, and implant design could have been a factor too, but now with 3D printing, you can do a lot more. And so what's the next step for those technologies? Can you coat them? I think it was mentioned earlier about biofilm. How do we make devices that resist biofilm formation or aid in infection prevention? With coatings, as technologies continue to improve, is this something that can be done with the 3D printer, or at least on top of coatings like that, or make them as vehicles to deliver that kind of stuff? These are all just way out there, things that could happen. They're not there yet, but okay, that's all I had. Thanks a lot.
Bem, então vamos lá, né? Eu vou falar em português.
All right, we will speak in Portuguese. Do you hear me?
E vou colocar o meu fone também para eu te entender direito.
I'm also going to wear my headphones so that I can better understand you.
Então, vamos lá.
All right.
Senhor Patrick, primeiro apresentação.
So Patrick Aldinger, thank you so much for your presentation. I think it was a very interesting approach, from the context and all the coatings of devices. So it's important for us to have this idea. Most of the presentations do not have that approach, and sometimes we do not really understand how it all came to be. We think that it's recent, but sometimes it, started way back then, and we've had a long road to get to the answers that we already have now as solutions. So considering your presentation and focusing, on the tests that are conducted to evaluate the coatings, the first question that I would have is, when you're evaluating the coating, we do not consider the device itself. So the technical standards require you to get the product and evaluate the performance on the product. So when we're assessing the coatings, that's not the same thing. In general, you get a coupon, and then you conduct all those requirement verifications. So in your opinion, is that enough? Considering that many products, like for example, the acetabular shells, you have those concave areas. So you understand that what is conducted on the coupons is representative for the final product.
Yeah, it does seem kind of not as representative, right, of the final device. You having a coupon that's nice and flat, you bring up the acetabular shell that's nice and conforming. There's hip stems that have a lot of features on them that, how do you know this is going to work well? Now, there's a couple issues, right, with trying to evaluate the final design. So I didn't get into animal studies, which is not really my background, but certainly those went on in the historical development of porous coatings to a degree, but those were animal models. They're still not orthopedic implants. And so to a degree that you're limited by what you can do to look at the final performance. But what we do is analog testing, right? That a lot of times we do analog testing just to show that, let's say you wanted to look at the strength of adherence, you could make a animal, a pin that's coated and you can push it out. It tells you whether or not it developed enough sufficient strength. So we're using this coupon level test to give us enough information that the coatings that we're developing are made well enough to perform when what's been benchmarked are all those historical stems from, 30 years ago. They were evaluated using these coupon-based tests, and we know that the stem performs well. We know with the value of the coupon, what in terms of value, I mean the parameter that it produces. So this coupon-based approach seems to be an accurate way to evaluate these coatings. Certainly with 3D printing, right? This is a new area. It was mentioned earlier that how do we know that this is going to work for those? There was a whole characteristic side of it that I said I wasn't going to cover because I don't know as much about it, but characterization of the pore structure itself, pore size, the spacing in between the pores, all that is critical. Their years were spent on developing what is the optimal pore size and porosity. So that's all part of those guidance documents.
Muito bom.
All right. At Anvisa, many times we are questioned about one thing. Inside the standard, there are many requirements that for us today, just to give you some context before, since 2008, our public rules in order to regularize an orthopedic implant requires for manufacturers to comply with the standards applicable to that device. And then in different events that we go to, people often question, if I do a fatigue test, for example, I wouldn't need to do the other ones because if I pass that one, I would be okay. Well, considering this context, if you had to choose among all the required tests to verify the characteristics and to see if they will actually result in a good outcome for the patient, if you were to choose just one test, like if the material passes on that one, the others wouldn't be necessary, which one would you choose?
That I described up there, but I pick one, I would pick all. That's just been the guidance across the number of years is these are the tests you have to do because they describe a different application for the coating, right? So tensile attachment is how well does it adhere to it. Shear fatigue, take for instance the example if you're just you're implanting this hip stem down the canal and you're rubbing it up against bone, you don't want it to shear off those beads. So all these different tests have some sort of application that they are addressing, that they're, whatever it be long-term use or whatever, those are aspects of the coating that are individually described in each of those tests. So I wouldn't just pick one. Like you can't just pick abrasion testing and saying, oh, well, it passed that test, it's not shedding off enough particles. Well, that's not the only test that matters for porous coatings.
Bacana. Bem, a gente pensa.
All right. We agree. We want it all. But sometimes we are questioned. Regulators always want all the tests, you know that. But it's good to get the same feedback from you as well. All right. When it comes to the validations that are conducted, especially when you have a combination of different coatings, we have received some examples of titanium associated with hydroxyapatite. So these combinations, do you see this as positive, as something that is actually bringing something extra to the end result? And what is the challenge when it comes to this combination of materials in a coating?
So you mean, what is the value of HA coating on top of another porous coating? Is that what you mean?
No, because you do an association of different coatings on the same device. Both coatings are applied on the same device. Because in practice, the coating was applied, but we used to use a single material, either hydroxyapatite or titanium. Today, we have devices that combine both of them. I want to know if you believe that that actually brings a better outcome in the end.
Yeah, so I can't really speak to how well HA was performing when applied on some of these porous coatings. I showed the clinical data on polar stem, which was a titanium coated, HA coated stem, right? And there's design aspects of that stem too that may lean it to being a better stem than other stems. But I think my understanding of HA coatings in general was it was more of a European mindset. They were shown to be effective in some cases, but it wasn't always super clear. So I think when and again, I might be botching the answer here, but from what I've observed when we developed hip stems is for the US, we just developed regular, let's say stick tight on top of R3. But then when it went to Australia or Europe, we would put HA on it because it was just a mindset of how HA promoted bone ingrowth. I'm sure it's founded in something because I mentioned it was developed in what, 1960 something, or it was being used in the 80s, I think is when it was first being used on orthopedic devices. So there clearly should be some benefit because why would you put cost into something that's not actually doing anything? There might somebody out here might know better about HA coatings and their effectiveness clinically, but.
Ok.
All right. So related to the number of samples, there's the FDA document that mentioned 5 samples, but in practice, the standard mentions 10. And we have faced some situations because in the end, manufacturers from the US, they follow the FDA guide. And in practice, Anvisa follows the standards. But without considering the standards or the guide, which one would be better or more right? But in practice, even if we have 5 samples, in your understanding, that would be enough for demonstrating? Is that enough of a sample size to give a final answer on what is being assessed? And if yes, why is the standard asking for 10? Considering that the manufacturers participate on the creation of the standards.
Yeah, that's a good question. So only one of the components of the ISO standard I think it's for HA calls out the 10, the sample size of 10. I'm not sure where that came from for that working group that developed that standard. I am involved in ASTM. I've seen some other people here who are involved in ASTM, and we go through these standards and talk about sample size. In that forum, we get a chance to also get understanding from the FDA because there's usually an FDA representative there too. And they have long considered that 5 or 6, mainly 5, has been sufficient to address most concerns with devices. Because you think about it, testing more would be great from a scientific standpoint. You could maybe understand a lot more, but the cost of doing a lot of tests for a lot of samples on full constructs becomes pretty cost prohibitive. So I think it was recognized not from a statistical standpoint, but more of a practicality and a cost to the implant that would ultimately be there if you tested 100. Now, I think it does depend on the industry that you're in. I'm probably going too far into something else, but like developing stents or something that is a different application, there might be a reason why more sample sizes are needed. You can go way off into the weeds on doing statistical analysis and how do you power your study to make sure you get this certain level of outcome. But I think one thing that we do is, and I think the industry as a large does this, that's why most of the ISO specs for orthopedic devices say 5 or 6, like the ISO 726-4 it does say minimum, but it says 6 runouts at a load. And so, I think there has to be a balance between what you're learning from that test. If it's a pass-fail test, is it just describing variable data? Are you comparing a predicate device to another one? One of the things you get into, which can be problematic, is when you compare populations and you show there's no difference, but your sample size is low, that can be problematic for regulatory bodies to understand. Like, it's not different, but let's say your average was slightly lower, but it's not different. Is it really different? Well, you didn't do enough samples to prove that it was really different. So I think my general answer is 6 samples is sufficient to evaluate most of the risks that we are evaluating. I was trying to think through some of the scenarios that I've observed, at least internally, where we tested 6 and the implant ended up not performing well based on that particular implant mode or failure mode, that test it wasn't because of a lack of sample size. So I think, again, balancing how much is the cost of some of these studies. Think about where studies. These are long-term tests with a lot of samples. If you want to push that up to 10 or 14 samples, you might get more information. Are you gaining enough sufficient information to change the risk level of this device? And I don't think it's there. So back to your point, I don't know why ISO came up with the 10. I'm not sure why I don't sit on ISO. I think Darlin does.
Acho que não, né? Alguém faz parte da ISO?
Anyone here part of ISO? Oh, yes, Professor. Oh, Darlin as well. Would you like to comment? I don't think he wants to say anything. He's probably going to kill me outside. Don't worry, Marcia. Well, the sample size is being discussed within the F04 committee at ASTM. There is an ongoing work covering, actually, to determine the sample size. And this is a topic that has been discussed for many years, actually, because the number of 5 for many cases is, or it sort of magically appeared in some situations. And we do have some cases for which we have evidence showing that 5 would not be enough. Some years ago, there was work to increase the sample size for vertebral spacers, but then it's a discussion that has been taking place. But I think that in the end, the way to go is determining the sample size that is representative for the result that you are assessing. I think this is the way that will be chosen at ASTM. It might take a little longer for ISO, but the idea behind increasing the sample size is to reduce variability and improve results and perhaps eliminate biases. Because we know with a larger sample size, you can have, or you can become closer to the average and avoid that small variations might put you further away from the average result. Just to comment on that, because Darlin and I participate on this discussion on the sample size at ASTM, and the discussion has evolved in a certain way, because it depends a lot on the acceptance criteria. So if you have conducted 5 coupons in a sample, and this is way above your acceptance criterion, the tendency is for you to reach the conclusion that your sample size is enough because your average is far from the criterion. But if you run 5 samples and you're close to the criterion, perhaps you need to run more samples so you have enough power to say that these results are above the criterion. So this is sort of how the discussion is being shaped, especially for shoulder replacement. They are already including that inside the standard. But it's this is the concern that Patrick has raised, that it is quite costly to conduct these tests. The products are very expensive, and historically, 5 samples have given us good enough results. So this is an ongoing discussion, and actually tomorrow I will discuss the development of new standards, and we're going to comment on that.
For these test methods and this guidance document that has been utilized for years, whether or not 5 was arrived through just magically or somebody actually had a good reason for doing 5, it appears to be sufficient to describe porous coatings as they exist today. Now, if you develop something new, a completely new technology, a new test method, I think it's probably worth evaluating the sample size discussion. But like ISO 7206-4, hip stem testing, I think if you make a stem that runs out at 5 samples or 6 samples at that load, the likelihood that it fails clinically is very, very low. So for existing standards and guidance documents, to me, it seems like we shouldn't challenge this is my view of it, of the sample size. But if you're going to develop something new, a new standard, a new technology that doesn't exist today, and you have to have a new test method like Professor Link mentioned, like non-standard tests, well, then you really need to evaluate the sample size discussion. But for consensus standards that have existed for decades, to me, I don't see the need in challenging those.
Joia. Obrigada aí, Darlan, Heitor.
Thank you, Darlan. Thank you, Hector. And thank you, Patrick. So I have one more question because we're almost out of time. Considering the changes and the new technologies, it has been discussed the importance of 3D printing and these new technologies, and the main challenge that we have to face in controlling the size of the pores on the substrate. So, from the materials that are applied, could you give us any idea on which materials could be used for future coatings, or would we stick to what we have today, like titanium hydroxyapatite? Is there anything being developed for the future in terms of changing the material or the nature of the material, perhaps a ceramic? Just thinking out loud here. I'm just sort of...
Yeah, so cobalt chrome is another one that's being looked at for sure. We internally at Smith & Nephew have other materials that we are evaluating for 3D printed applications. Ceramics, I don't know if you can do it that way because the way that it has to be finalized. I don't know if it's possible. Maybe it is, but those are the only two I'm aware of, except for the ones that we're developing internally. We're probably most likely going to continue to use titanium and cobalt chrome because that's what we've used for decades, and they work well when they're designed appropriately, like Professor Link said. So those are the best biocompatible materials we have. And so unless somebody develops some new future alloy, and over the last 30 years, people have developed different variations of titanium alloys that aren't used today. For some reason, Ti-64, Ti-67, cobalt chrome, those are all kind of the materials that we continue to use. So yeah, I'm sure there is, as people gain more understanding of how to produce and manufacture 3D technologies, as the manufacturers of the equipment get better, the materials that you can process with them will probably get better as well. So I imagine at some point it'll probably get better, or you'll end up seeing other materials.
Muito bom. Bem, gente.
All right. We have 3 minutes left, so I don't think we have time for answering any more questions. Oh, I'm sorry, Hector. Sure, go ahead. I'm sorry. I actually have a question. It's quite a quick one. You mentioned that it is important and necessary to do all the standardized tests to assess coatings. My question is related to the fact that we see a lot of combinations. You have cobalt chrome, pure titanium, different titanium alloys, and then we have different coatings, hydroxyapatite, titanium, titanium with hydroxyapatite. So my question is this: do you do any type of worst case analysis for these combinations? Because if we think about it, different combinations, they generate an endless possibilities if you're going to combine all substrates with all coating options in all the assays. So my question is, do you do all combinations, or do you use a worst case analysis in which one combination can represent others?
The combinations of the devices we would make. So like if I don't think we do HA on a cobalt chrome tibial component or femoral component, at least we don't. So that might be worst case for some reasons, and some people might offer that, but I haven't evaluated, but that would make sense. Whatever, I mean, any multi-coated surface you have, you need to do these tests, and this is the book of tests that we have to evaluate coatings. And so, yeah, you should certainly look at all the different options you plan to market, I would say.
Ok. Mais alguém? Minuto.
Anyone else? We have 1 minute exactly. Go ahead. making the most out of your time, Patrick. Good afternoon. With the development of nanotechnology, we started to see some coatings with a nanometric thickness. Despite the fact that that is very good because it's compatible with the size of stem cells, we do have a serious issue in including the validation of this nano coating if you follow the validation pathway that you presented. So my question is, what is a possible validation process for that? Because pretty much none of these assays are applicable to 50 nano or 25 nano thickness. And how can regulators, FDA and Avisa, how are they going to perceive this new validation?
That's a good point and a good question. I don't know. Because I've never looked at that technology, I don't know how I would try to tackle it, I guess, from a testing perspective. I welcome anybody else who has an idea because, I mean, yeah, the testing that we do is more of a, I would say, the approach that's taken with these coupons and an adhesive film is a little bit more on a macro scale. That adhesive is not telling you anything about the tiny little pores that could exist. It would be interesting that, you know, some of these asymmetric powders that were also very porous, what they did to show that it worked fine. It could be they did the normal gambit of tests, it passed those, FDA said fine, and whether or not they work well or not, if that nanostructure is actually doing anything, I don't know how you would. That's a very good question. You'd have tiny little coupons. I don't really have a good, very, very nano-sized coupons with nano equipment to test it. I don't. It's a good question. I have.
Eu? Poxa, aí você me matou.
Myself? No, come on. No. There's no way out of here, I think. No, it's actually because we don't have more time and we are out of time. And once we run out of time, we have to leave. You know? So like the Oscars, when they start playing the song, you have to leave the stage. So, Darlan, I'm not going to go anymore, okay? But anyway, all jokes aside, there are some things that we don't know about. I mean, we have innovation, new types of coatings, new types of products, new designs, and it is a challenge for everyone, even for us from the regulatory arena. We talk to other regulatory authorities and we all face the same issues. There are some things that do require an answer, but we struggle with finding them. And based on what we learn, we try to find a way to solve them together on how to approach those issues. But many times, even one of the questions that we got that we didn't have time to ask, but one of the questions mentioned the fact that we could use comparable from a different manufacturer to assess a certain requirement when you don't have a reference. In some cases, you can do that depending on the level of information that you have, but in other cases, that is not a possibility. And in practice, this is what we have to deal with. Every new technology poses a challenge to everyone. All right. So I think that was it. Thank you very much, Patrick. I know it was difficult for you to get here. You almost didn't make it, but in the end, it all worked out, and we are glad it did. Caroline, thank you for the effort in making sure he got here. It was great. I had many more questions, but unfortunately, we have to go. We are all tired. We have a big day tomorrow, and it's never easy. But Dr. Sergio, thank you very much. It has been great. And like always, this is already a hit. I think everybody who has presented today were able to share important information that will make us think about and perhaps rethink everything that we do. So thank you very much, and perhaps see you tomorrow. Can I say that? Or is he the one that gets to say that? All right. We also would like to say thank you to Patrick Aldinger and Ms. Marcia Ribeiro for their participation on our seminar, a seminar that is sponsored by Vim and Sistemas. I would like to request all of you to use the same QR code to enter the building. So make sure you save your QR code. And now I'd like to officially close this event. And to do that, I'm going to invite Mr. Sergio Madeira for his final remarks and Mr. Augusto Geyer, General Technology Director for Health Products at Anvisa.